Afternoon. Afternoon. Any questions before we get started? I know I have two questions that I want to address, but any other questions before we get started? I'll tell you what, let's address the first two questions first. First question was a blood vessel question about the fibular vein. The fibular vein, or again, also perennial vein, those two terms are interchangeable. I would say primarily feeds into the posterior tibial vein, just prior to entering into the popliteal vein. Again, one of the things you have to remember is there is a fair amount of anastomoses, a lot of lateral and collateral routes for these. So it isn't a straight shot, but I would say that, so if you went straight, if you were given driving directions, you went straight to the popliteal, I probably wouldn't ding you on that. But I think it's more accurate to say that the posterior tibial vein, the fibular vein feeds in the posterior tibial before it goes in there. What I mean, you know, like anytime you're at, like, you have like a raffle or something like that, they talk about how you have to be present to win for that. And that's definitely the case on Thursday. On Thursday, as I've mentioned, we are going to be forming our groups and for those 50-point presentations, and then those groups will be selecting their topics. And again, you must be in class on Thursday to be able to participate in this, to be part of the group, because we're not just forming the groups and selecting topics, but you're going to have time to work on this starting on Thursday. So this is an important project that you guys are going to be working on together, and to make sure that it is fair for everybody in the group, I want you guys going to be sharing the workload on this, and so I want to make sure that everybody is there on those days to be part of it. So obviously there's nothing to win. It's just like anytime you go to like one of those raffles or something along those times, you know, it says you have to be present to win. So that was the point. Be there on Thursday or you lose out on 50 points. So that's for that. Nope, you get to pick your topics. I will give you the categories from which you can choose from, but the groups as a group will decide what topic they want to use. And then as long as there's only going to be one topic per group, I don't want three groups doing the same topic. And then you will get to select your topic. You'll give me your top 2 or 3 topics. I will try to make sure that everybody gets one of their top 2 requests, and then we will pick them, and then you'll start working on those right away for that.
How many people per group?
I'm sorry?
How many people are going to be in each group?
2 to 3 people. So depending on how many people shows up, the groups, again, you can't have a group of 1. So, I find if the groups are larger than 3, then it is too easy for not everybody to carry their weight in that size group. So the groups will be 2 or 3, depending on how many people survive this first exam and show up on Thursday, we'll decide the groups. But the groups typically are between 2 and 3. This is not a new activity. This is something I've been doing for a very long time in 431. This will be the first time we're trying to do it online, so that's going to be interesting to try. But I think it lends itself to this format, and so it will be okay to try to accomplish it in this fashion. All right? Because again, you're not being graded on your presentation skills, and we'll talk about this a lot more when we get to it. You're not being graded on your presentation skills, but you're being graded on the information that you provide for us. So as long as you're providing that information with a visual cue by sharing your screen or doing something along that lines, then that should meet the needs. But like I said, we'll talk about that on Thursday when we come back, but we got a lot to do before then. Well, not a lot, but we still have some things to do. We need to finish up our discussion of blood. We have some more blood physiology to talk about. And then for our lab, even though you've done blood typing, no, you cannot take the test before 12. Blood typing as a lab star activity, or one of your physio X's was, which one was it? Yeah, lab star, I think, was the blood typing activity. We're still going to talk about it, make sure that we all understand that, because that's an important concept. And there should be still question times for a question and answer review for that. The exam is going to be next Tuesday, and again, it must be taken during class time. That means you cannot take it before noon. You cannot take it after 4:35. And again, the key I emphasize on this is it must be completed during this time. If you wait till 4 o'clock to start your lab exam, then at 4:35, your lab exam shuts down. So you will only have 35 minutes to complete it. So you want to make sure that you don't do that. I will be randomly, I will allow Zoom. Zoom can do breakout groups. So what I'm going to do is based on the number of people, I will set the number of breakout groups and it will randomly distribute you into groups. I know some of you have the advantage of knowing others, but other people don't. So I think the fairest way to do the groups is just to have it randomly assigned. So since we're doing this on site, we will be randomly assigning the groups. Again, none of this matters because it's not till after the exam. So let's not worry about the groups. I know everybody hears group presentation, everybody panics. It doesn't matter. Guess what? You're still taking a lecture exam. It's still going to be worth 100 points. You're still taking a lab exam. That's going to be worth somewhere 60 to 70 points, right? If you think about it, you've had more than 50 points of homework during this section. So this is your 50 points of homework during this section. So it is not something you need to be concerned about, and definitely something that you don't want to worry about until the 17th when we talk about it, all right? So let's talk about the exam. Again, it must be completed within the class time. I have not finished your exam yet, but this exam typically has 70 some questions. So my guess is it's somewhere around 70, probably somewhere between 70 and 80 questions. There's a fair amount of anatomy on this one. I will tell you from experiences from last semester when we were forced online, but also summer when I was teaching fully online, most of the problems people have with their exams, they have with their lab exams. You are getting 70 some questions that with high resolution images that all have to load on your screen at the same time. And so some people have challenges with that from their computers. So the two important things to remember with that is I encourage you to take your lab exam first and to take it as close to noon as you possibly can for that. So if you have problems, you have time to resolve those. Also, if you have problems, if the computer locks up, if Canvas locks up while you're trying to do it, if you get kicked out of the exam or something like that, then you contact Proctorio's support. There is a Proctorio link that you click on, and they are the ones that can get you back into the exam. It doesn't do you any good to reach out to me, hey, I just got kicked out of the exam, because I can't do anything about that. I do not have the means to allow you back into an exam. Only Proctorio tech support is able to do that. They're able to do that, but those are the people that you want to contact. And remember, if you get kicked out of the exam or your exam freezes on you, the clock keeps running. So it is important to make sure. So yes, if you lose internet service, which hopefully you won't do, but if you do lose internet service, as soon as you get it back, contact Proctorio. You may try to get back in on your own. Sometimes if you lose internet service, I've had students who were able to get back in on their own when they've been kicked out. But if you can't get back in, you contact Proctorio's support. They will get you back into the exam. I cannot do anything about that. All right, so they're the people that you want to contact if you have that. But remember, your timer keeps going. So if you decide to go eat a sandwich before you try to get back into the exam, then you're losing a half an hour of the time. Yeah, there you go. Excellent. Ashley had that problem last semester and was able to get back in. All right. Again, lab exams vary points wise. So again, and I haven't finished yours yet, but my guess is usually it's somewhere around 90 minutes for a lab exam at the most, usually less than that, but 90 would be the most. The lecture exam, again, usually somewhere between 30, around 30 multiple choice questions, somewhere between 2 and 6 fill in the blank. And then about 7 to 9 essay questions. Depending on the length of the exam, it'll be somewhere between 2 and 2.5 hours that you will have to complete the lecture exam. So if you think at the most 2.5 for that, at the most 1.5 hours for your lab exam. So that's 4 hours of exam time that is available to you. Again, you're not required to use that time. If you finish the lab exam in 30 minutes, then that's totally fine. If you finish the lecture exam in 1.5 hours, that's totally fine. In fact, most people will not use the full time to be able to do that. But you want to at least give yourself that time if there are problems or there are issues or if there are things along those lines. But notice with that, even with that, it's 4 hours. So I encourage you to start as close to noon as possible, take the lab exam first, and then take a break. Take a 15, 20-minute break, right? Get something to eat, get something to drink, go to the bathroom, cry, whatever it is that you need to do, and then start the lecture exam and complete that as well. It varies from test to test. My guess is, again, I haven't finalized this test, but probably usually it's between 2 and 6, fill in the blank. Those are usually like vocabulary and things along those lines. I shouldn't have to give you an idea of what the essay questions are. Being the sophisticated 431 students, you should be able to do that. You should be able to look at the things that we've talked about in class and say, well, let's see, what are some of the big physiological processes that we talked about? Oh yeah, we talked about the cardiac cycle. We talked about the cardiac conduction system. We talked about the action potentials of conductive and contractile and skeletal muscle cells and compared those and all those types of things. You should be able to go through those. Again, one of the things I've always loved about this class is hard. I don't have to be tricky. If you've had a process that you've talked about in class, or you've had a homework assignment about, or there's been another activity, or like, I don't know, maybe the ECG, right? Do you think that there's likely to be an ECG question since you had an interactive physiology activity on it, and that had a big homework assignment that went along with it, and we talked about it in lecture for an hour? I mean, those are all the things that kind of are important. You should be able to look through the lectures, look through the slides, figure out the things that are important, and those things that are important are typically the things that show up on an exam. All right. All right. Any other questions on that? Again, we just talked about the test. I usually do that during the review, but again, we should have time for that as well. Remember, review isn't me standing here telling you what I think is important. A review is your opportunity to ask me questions about things you're unclear on, and then we'll work together to make sure you understand those concepts. Right? I tell you what I think is important every day in lecture, so I'm not going to recreate that. And remember also, if you don't ask questions, I've assumed you've all mastered the material, and I make the tests harder. All right? Questions on any of that? All right, there was one other question that was asked at the end of last class, and it had to do with your anatomy list. One of them was, what is the moderator band? Someone tell me what the moderator band is. I will wait. What is the moderator band?
It's in the right ventricle.
Okay, that's a good start.
A big piece of muscle.
Okay, that's excellent. What's its function? Do you know? Okay, excellent. So you've absolutely got the right idea. As it turns out, in our sheep, and again, normally you got to remember this list was put together when we were in the classroom. On the second day of class, we do a sheep heart dissection. And part of that sheep heart dissection is exploring all of the chambers of the heart. Remember, one of the things we talked about is that the left ventricle has a lot of advantages over the right ventricle. It has a thicker muscular wall that allows it to produce a more powerful contraction. It has that circular organization to it, making it very efficient in its pumping. And that poor right ventricle is thinner in-walled. It wraps around the left ventricle so that it doesn't have as efficient of a space. Again, as we talked about, if you make a fist with your hand and squeeze that, and then you put the other hand around and try to squeeze that, it's not nearly as efficient. But we do know that the right side has to pump the same amount of blood as the left side. So we need to give it a little bit of an advantage. And it turns out in our sheep hearts, there is a thin band of muscle where some of those conductive cells, some of those Purkinje cells, or actually really I guess it would be bundle branches, where some of those bundle branches that normally go down towards the apex of the heart instead are able to cross over to the wall of the ventricle. It's basically a shortcut for the electrical signal to get to the right ventricle to help in the efficiency of the right ventricle's contraction. And every single sheep heart I have ever opened up has always had one inside of it. The real question becomes whether or not humans have these moderator bands. And it is unclear. In fact, again, normally when we're in the classroom, one of our class models does kind of a half-assed job of putting one in there, and the other one doesn't have one in there at all. So it is a very obvious structure in the sheep heart, in the right ventricle of the sheep heart. It is not as easy to see in the humans, and there are many that debate whether or not humans actually have them. One of the differences between sheep and humans, obviously, is anatomical position. We stand upright, whereas the sheep is horizontal in its anatomical position. So again, it needs more help to be able to pump that blood to the lungs because of the orientation of it. So it's something that is only seen in the sheep heart, and I only show it in a sheep heart. And again, since we didn't get to do the sheep heart dissection, the only way you would see it is on a picture of a sheep heart. But those are things that you could see, and so I would encourage you to take a look at that. It is a fairly obvious muscular structure that goes from the interventricular septum to the wall of the right ventricle. Someone also asked what the auricles are. What are the auricles? Can someone remind me of what those are?
I know that they're on top of the atria. I don't know what they do.
They're the edges of the atria.
Yeah, exactly. I'd be careful on top. You absolutely have the right idea. They are the edges of the atria. The atria, remember, as we saw on a real heart, and again, let's actually look at the real heart. If we actually go back, let's do that. I need to get to the right place. I think this is where my real heart picture is. There we go. Again, I keep drawing that schematic of the heart as a big circle cut into fours. But as we saw when we first talked about the heart, the ventricles are these big, huge, massive structures, right? Here is, that's not showing up very well. Let's see if Blackboard shows it better. Here is our right, whoops. Here is our right ventricle. Here is our left ventricle, much, much larger in its structure, right? And then here, this tiny thing right there is the left atrium. This tiny thing here is the right atrium. The atrium isn't really a full rounded chamber. It's really the muscular wall that's kind of flattened. So that what happens is it fills up and then it pumps and it fills up and it pumps and it fills up and it pumps. And so notice that because of that, it has this kind of ruffled edge where the two pieces come together. And that's actually what auricle means. Auricle means dog-eared. So if you look right here at the edge, we have this dog-eared appearance, and that dog-eared appearance to the atria, where the two walls of the atria come together. And so that roughened dog-eared edge, that tip of the atria, that is the auricle. All right?
But both atria have it, right?
I'm sorry?
Both atria have it.
Yes, both atria have auricles. That is correct.
And you say it's on the edge, not on top?
Yeah, I would say it's the edge. It's the edge of it, yep.
Okay.
Yep. All right. Any other questions on any of the anatomy? I wouldn't say that the auricles necessarily have a function. It's a great question. I think it's more just an anatomical feature of the way that they're built. So I don't think it necessarily has a specific function. Maybe it helps in the efficiency of the pumping, right? Because then it makes it kind of like a fulcrum that it, but I think it's more just an anatomical feature. Yeah, being flabby or being collapsed, sure. Yes, it's on your list. Again, it's one of those things that I feel bad about, obviously holding you responsible for a sheep heart when you've never actually had the chance to dissect it. But remember, if you were taking 430 right now, and some of you did over the summer, the people who took 430 over the summer had to learn everything about the bones, all the bone features, and they never got to hold one of those in their hand either. So yes, unfortunately, there are plenty of pictures of moderator bands you can find online. Do sheep heart moderator band as a search. It's a very obvious structure that can easily be found. And yeah, it's on your list. It is something you do need to be responsible for. Again, I apologize for that. I hate the fact that the process gets in the way of the learning in this, but as I keep saying, unfortunately, that's the necessary evil of us dealing with this unique situation that we're dealing with. All right. Any other questions? Oops, too far. We're kind of getting our review out of the way now, but I'm okay with that. All right. Let's finish up what we were talking about in the last class then. In the last class, we were talking about the process of hematopoiesis, that process by which we make all of the formed elements of the blood, the red blood cells, the white blood cells, and the platelets, right? Or the erythrocytes, all the leukocytes, and the thrombocytes. As we talked about, every single formed element has a primary stem cell. And how many primary stem cells are there for all of the formed elements? One. Hemocytoblasts. Every single formed element has a secondary stem cell. And how many secondary stem cells are there?
Two.
Two. That's convenient. One primary, two secondary. That is certainly convenient. And again, these are stem cells. All three of these are stem cells. They're cells that don't have any other job other than to divide to produce new cells. The hemocytoblast and the myeloid stem cell are pluripotent, but the lymphoid stem cell is unipotent. And so if you form a lymphoid stem cell, how many possible different cells can you produce?
One.
Just one, just the lymphocytes. Excellent. And of course, what determines what type of secondary stem cell the hemocytoblast becomes, or what type of progenitor cell the secondary myeloid stem cell becomes, and what directs the directionality of this process? Chemical signals
or hormones.
Primarily hormones, but other chemical signals as well. Excellent, excellent, excellent. Perfect. All right. Now, while I said a lot of that, great question. Really, okay, so let's say it this way. There are ways to distinguish the secondary stem cells both from each other and from the hemocytoblast, but that is a level that I'm not holding you responsible for. So if you've looked at your histology list, while you need to know the formed elements, the mature formed elements, I will not be, yes, there was no daily quiz today. I will not histologically show you one of these three cells and have you try to figure out what they are. So instead, what will happen is I will show you a erythrocyte and ask you, what is the primary stem cell of this formed element? What is the secondary stem cell of this formed element? And so I'd ask it that way. I'm not going to show you a myeloid, a lymphoid, or a hemocytoblast and ask you to identify it histologically. I guess technically I could show you a graph like this without the labels and ask it, but that's not how I'm likely to ask it. I'm more to show you the mature cells and ask you what they came from. All right, so as I mentioned, you don't need to know most of the pathways involved in this process, but the one that I do want you to understand and be familiar with is the process by which we make red blood cells. And the specific process by which we make red blood cells is erythropoiesis. So this is our process of making red blood cells. Excellent. So once we have that, let's start simple. Primary stem cell. In the process of erythropoiesis, what is it?
Hemocytoblast.
Excellent, hemocytoblast. Secondary stem cell.
Myeloid.
Excellent. Myeloid stem cell. Excellent. Perfect. So that is our starting point. We go from that hemocytoblast to a myeloid stem cell. And remember, both of these are pluripotent. And what's the key to being a pluripotent stem cell?
Can turn into multiple things.
Excellent. There are multiple things that they are able to become. However, one of the things that a myeloid stem cell can become is what we call a committed cell. A committed cell basically means at this point its path has been determined. And that committed cell, as you can see from the illustration here, is a pro-erythroblast. Now, this, oops, again, helps if I spell these things correctly. This pro-erythroblast is a unipotent stem cell. Right? Again, it is still a cell that doesn't have a job other than to divide to make new cells. However, at this point, every cell it produces is all going to become one thing and one thing only. All cells produced will become our erythrocytes, our red blood cells. So at this point, the cell is committed. Right, at this point, there aren't any chemical signals that can change the outcome of what's going to happen here. We can speed it up, and we'll talk about how we do that in a minute. But at this point, the path is committed. All right, so these are the three stem cells related in this process. And notice as we move from this, the first thing that forms is an early erythroblast. And as you look at this early erythroblast, does it look a lot like a red blood cell as we know them?
No.
No.
It's purple. Because it's really purple? Is this cell really purple?
No.
Yeah, actually turns out it is. I know color is one of those things that we beat into your head that you shouldn't pay attention to. But remember also, there is sometimes, like with adipocytes and things like that, where color actually does matter. And this is one of these cases. Notice that these early erythroblasts have a huge nucleus and a dark purple color. The reason it has this huge, massive, dark purple color is because the cell contains massive amounts.
Ribosomes?
Well, yep, absolutely. And where are most of the ribosomes located?
Reticular,
Where is most of the red blood cells?
rough
Reticulum.
endoplasmic.
But you're absolutely correct. Massive amount of ribosomes, massive amount of rough endoplasmic reticulum. And of course, what do we use that rough endoplasmic reticulum for?
Make proteins.
Make proteins. Excellent. And specifically, what protein are we going to make here?
Hemoglobin.
Exactly. The cell starts making hemoglobin. Excellent. So it first starts as this erythroblast, as a big protein-making machine. And notice, as it transitions from early to late in erythroblast, the color starts to change. The cell becomes more pink and might start taking on more of a pinkish or reddish coloration to the cell. What does hemoglobin do?
It attracts oxygen.
Yeah, exactly, absolutely. What happens is, as more and more hemoglobin is formed, the hemoglobin starts to bind more and more oxygen. And as we know, when the oxygen binds to that iron of the hemoglobin, we start to get a reddish color, and the cell gets more pink. All right. So as the cell is making more and more and more of this hemoglobin, it's taking on more and more and more of a pinkish coloration to it. And it is filling more and more with hemoglobin. Now that is the one thing I don't like about this picture. As you look at this picture, it looks like as we go from left to right, that the nucleus gets smaller and smaller and smaller inside the cell. Really what's happening is the cell is getting bigger and bigger and bigger in this process. So the relative size of the nucleus gets smaller, but this nucleus isn't getting smaller. It's not like it's using up its DNA or something like that. The cell is just getting bigger as it accumulates more and more hemoglobin. All right? So far, so good? Questions on this? I'm gonna cheat and move my arrow here so that it's out of my way so I can write a little bit more. All right. The cell continues to accumulate hemoglobin until it reaches a critical point. Where it has contained a massive amount of hemoglobin. And when that occurs, the cell becomes a normoblast. So we'll get to that as well. Oh, great question. Yep. So that's exactly what we're going to talk about next. At this point, as a normoblast, the cell undergoes a cell process where it ejects its nucleus. Now, we've talked about cells that eject their nucleus before, right, as part of a death occurring process. It chucks its nucleus, it chucks its organelles, right? It destroys its DNA in this kind of death throes process. And what do we call that self-programmed cell death?
Apoptosis.
Apoptosis. That is not what's happening here. This is not apoptosis. The goal of the cell isn't for the cell to die, at least not right away. The cell's not going to die right away, but you guys have the right idea. The cell, the accumulation, reaching that critical point, a critical mass of hemoglobin, triggers the normoblast to eject the nucleus and most of its organelles. Now, again, remember the question was, where does it go? Remember, these cells are located. Where is this occurring? Where does this whole process, let's actually go back. That's actually a great question. Where, oh, no, hold on. Too late now. Where does this process occur? In the red bone marrow, excellent. So when its nucleus gets ejected, when its organelles get ejected, that gets broken down into bits. Those bits get reabsorbed by the stem cells that, like the hemocytoblasts and things like that, that are made and are used to recycle and make more cells. Because after all, if you're going to divide rapidly, you need to keep dividing your genetic material, and so you need more resources to be able to do that. So yeah, so the resources, this nucleus and these organelles, when they're released, are just going to get recycled by the stem cells that are there in the red bone marrow. Now, once it reaches this point where it ejects the nucleus, it becomes a reticulocyte. And it is actually as a reticulocyte that it actually enters into the sinusoid capillaries and enters the blood supply. So it is actually as these reticulocytes, which are essentially immature red blood cells, that it actually enters into the blood supply. In fact, if you were to draw out your blood and look, in a normal, healthy individual, about 1.5% of all of your red blood cells are reticulocytes. And so, of course, then 98.5% are going to be mature erythrocytes. And then after a day or two in the blood vessels, the cell matures into its mature shape, size, and shape, and becomes a erythrocyte. So notice it is ejected as the small little anuclear ball into the red blood cells, and then eventually forms that biconcave shape that forms our red blood cell. And again, how long does that erythrocyte last for?
120 days.
Yeah, from 100 to 120 days. Excellent. So notice we constantly are losing red blood cells, so we are constantly making a large amount more. Again, I've written it all out here, but let's look at the pretty pictures. Again, we start with that hemocytoblast, form the myeloid stem cell, form that committed cell. And again, that committed cell is a unipotent stem cell. At this point, there's one thing and only one thing it can become, a red blood cell. It is then going to actively produce hemoglobin until it reaches that critical mass. At that critical mass of hemoglobin, it ejects its nucleus and becomes that reticulocyte. And like I said, we are constantly losing red blood cells, so we are constantly making more. About 2 million per second. So every second, you have 2 million new reticulocytes entering into your blood supply. Massive, massive production process going on here because you're constantly losing massive amounts of red blood cells as well. Absolutely, yes. So the question was, so do they enter all the different areas to the blood where the bone marrow is? Exactly. Anywhere there's bone marrow, there's going to be those sinusoid capillaries. Let me rephrase that. Anywhere there's red bone marrow, there are going to be sinusoid capillaries where we're going to be making these reticulocytes and releasing them into the blood supply. So yeah, some are being made in your legs, some are being made in your hips, some are being made in your skull, all over the place. Sinusoid capillaries are in the bone marrow. Remember, bone marrow is one of the places where we find them. The liver is one of the places we find them. Some of the endocrine glands will have those sinusoid capillaries. Remember, those sinusoid capillaries were those big, loose capillaries where the epithelium isn't complete. So there's big holes, there's big pockets in it. So something large, like a reticulocyte, can get in and out. The spleen and the liver are places where we have the the sinusoid capillaries, because that's where the dead and damaged red blood cells can be pulled out of the blood supply so that we can recycle the iron, we can recycle the resources from those in those areas as well.
What did you say the difference was between a reticulocyte and just a normal red blood cell?
I guess the two main differences would be their age and their shape. Right? They're only a reticulocyte for the first couple days after they're released before they mature into it. And again, they're only a small fraction of the blood supply. And as you can see, they're small ball-shaped cells instead of that unique biconcave shape that it has. So I think I said a small ball-shaped anuclear cell, because obviously it doesn't have a nucleus, but it then forms that biconcave shape of the erythrocyte.
Okay.
Now, normally we don't distinguish when we're doing those hematocrits and things along those lines, but if you're 3 weeks out of surgery and I take your blood supply and I look at your blood supply and I see that you have 4.5% reticulocytes in your blood, what could that be an indication of?
Bleeding.
Internal bleeding, exactly. If you are losing blood, then obviously you have to replace it. You're going to be making a lot more, and your levels of reticulocytes will go up. Now, obviously, right after surgery, when you've lost blood, you expect it to be high. But if 3 weeks later you still see a really high reticulocyte count, that could be an indication of some type of internal bleeding where they're still losing blood and having to make more. Exactly. So knowing how many are in there can be an important. Well, again, if you think about it, to maintain homeostasis, we want to be able to make blood cells at the same rate that we're losing them. So while if we're making 2 million new red blood cells every second, then my guess is that we're probably losing or dying, or 2 million per second are dying or being lost. So that would be my guess, because we want to maintain homeostasis. All right. Questions on that? All right. Now, as we mentioned, this process of erythropoiesis is controlled by chemical signals. Oh, exactly. If you do not make enough red blood cells, then you are considered anemic, right? Remember, you're considered anemic if you are not able to get enough oxygen to the tissues of your body. If your tissues of your body become hypoxic, anemia could be a cause where you don't have enough red blood cells, or your red blood cells don't have enough hemoglobin inside of them because you're lacking iron, or maybe you produce non-functional hemoglobin subunits or something along those lines. But as we also talked about, circulatory problems with the pumping of your heart could cause it. Problems with the blood vessels could cause it. But as we also talked about, going up to Vegas for the weekend, right, or Tahoe for the weekend, right, being at that higher altitude where when you inhale a volume of breath, it doesn't have the same amount of oxygen in it. All of those things are things that can be a warning sign that the tissues aren't getting enough oxygen. And as we talked about, this is going to be monitored in the kidney. At rest, as we talked about, at rest, 25% of the blood travels to the kidneys. So it makes sense that the kidneys would be an important location where you measure the oxygen carrying capacity of the blood. So kidneys measure the oxygen carrying capacity of the blood. And if it sees that the oxygen carrying capacity is low, then our kidney will produce the hormone erythropoietin and release it into the blood. And of course, what is the target of erythropoietin? Bone marrow, excellent. Red bone marrow. And that erythropoietin does two things. The first thing it does is it causes the primary and secondary stem cells to form more proerythroblasts. Right? Getting more committed cells. What this is going to mean is we get more red blood cells as a result of that, because you're going to make more red blood cells than that. But the other thing it does is it causes the proerythroblasts to make hemoglobin faster. So the effect of that is that the cells mature more rapidly. So notice, not only do we make more red blood cells, but we make more red blood cells more quickly. All right? Questions on that? So again, we get more proerythroblasts, and those proerythroblasts develop into reticulocytes more quickly. And your book's got one of these pretty teeter-totters that it likes to use to show that imbalance. Right? Oxygen carrying capacity goes by, sensor is located in the kidney, produces a hormone. That hormone goes to the red bone marrow. That red bone marrow increases production of red blood cells, and oxygen carrying capacity of the blood goes back. All right, questions on that?
Do you want us to remember all the proteins names?
What do you mean?
Spell them like.
Yeah, do you need to know how to spell erythropoietin? Yes.
Okay.
Hemoglobin? Yes. Hypoxia? Yes. Proerythroblast, normoblast? Yes. Spelling matters. I don't care how you pronounce these things, but spelling matters. All right, questions on that? All right, with that, we are done with our red blood cells. We need to talk about our white blood cells, or our leukocytes, next. All right, again, we're not going to go in quite as much detail. The reason for that is we'll also talk about these cells more when we get to the next section and we're talking about our immune response. But we need to at least identify the cells, talk about the cells, and understand their basic functions, at least at this point, as well as being able to recognize them. We need to be able to do that as well. They're far, far less numerous than our red blood cells. Remember, as we talked about, red blood cells make up over 99% of the formed elements. So remember, while that single microliter of blood has 5 million red blood cells in it, it only has 5,000 to 10,000 white blood cells. Now, one of the important things to remember is that doesn't mean that you only have 10,000 total white blood cells, because at any one time, only about 2%, again, assuming you are healthy, only about 2% of your leukocytes are actually in the blood in circulation. The rest of them are kind of chilling and hanging out, places like our lymph nodes, places like our skin and our lungs, within our lymphatic fluids, within our spleen, within our liver. However, if you become sick, can those cells be mobilized and brought to your defenses and those levels go up dramatically? Yeah,
Yes.
absolutely. So yeah, so as long as you're healthy though, most of them, again, it's like the Army, right? At any one time, a couple of soldiers are out on patrol going around the perimeter, but everybody else is hanging out in the barracks, hanging out, you know, in the cafeteria, right? You know, playing basketball or things along those lines. But when there's danger, everybody's mobilized and brought to the party. Now, as I mentioned, there are 5, and let's do this up here just to remind us, 5 main types of white blood cells, or again, leukocytes. And as we talked about in the last class, they fall into one of 2 categories. The first categories are our granulocytes. Yes, cytokines are one of the major cell-to-cell chemical signals. So they're like a local hormone that is used to mobilize the white blood cells. They also help us to determine which of the 5 types we're going to make. So depending on what type of pathogen you're dealing with, those cytokines are going to determine which of our types of leukocytes we're going to want to mobilize and which ones we want to divide. And yeah, absolutely. Cytokines, for those who don't know, are basically cell-to-cell local chemical signals or local hormones that are used by cells locally to communicate and control much of our white blood cell production and development. And like I said, we'll talk about that more. It's one of those fun things. We will talk about it in when we get to our immune response in the next section. For those of you who have had 430 already, you're going to see that there's a lot of similarity in those things we talk about. And so one of the nice things for me is that I know I can glaze over some of these things because I know you've gotten it or will get it in micro. So for those of you who had micro, might get a little bored at times because we simplify things a little bit. Those of you who haven't had micro might be a little terrified by the things that we talk about. But between the 2 classes, you'll get everything you need to know. All right. 2 flavors of leukocytes. The first are our granular cytes, or granular cells, called so because of the large vesicles of chemicals that they contain inside of them. The most common of all of our granulocytes, and in fact the most common of all of our leukocytes, are the neutrophils. Notice the neutrophil is slightly larger than a red blood cell. And there are really two key characteristics that are going to help you to be able to define it. What do you notice about its nucleus? How many nuclei does it have? Anyone know how many nuclei it has?
More than one, for
It
sure.
has one nucleus.
One.
But as you can see, that nucleus is multi-lobed. So it doesn't truly have two nuclei or three nuclei. What it has is a single nucleus that has all these different lobes to it. So it looks like it might be multinuclear, but they're all actually interconnected. So it actually has what we call a multi-lobed nucleus, where it looks like it has multiple nuclei inside of it, but they're all interconnected. So it is truly just one nucleus. All right, so we have that multi-lobe component to it. The other thing it has is fairly distinct granules. You can see, and again, my dots there are way bigger than they need to be. Is that any better? That's not bad. We see the dark granules that are fairly distinct, fairly easy to see, but not so much that they're blocking the nucleus. We can clearly see the nucleus. We can clearly see the cytoplasm. As I mentioned, these neutrophils are the most common of our leukocytes. These neutrophils are primarily our first responders. Most of our white blood cells have this characteristic, but in particular, our neutrophils, and one other we'll talk about, are our phagocytes. Someone remind me what a phagocyte is again?
Big eater.
I'm sorry, say again, Susan?
Big eater.
Exactly. Well, okay, they're not the big eaters, but you have the right idea.
Yeah,
A
they're the ones that.
phagocyte is a cell that eats other material. Their job is basically to engulf extracellular material, things like viruses, things like other pathogens, abnormal cells, dead, damaged cells, things along those lines, and engulf them, break them down. Part of it is being the cleanup crew, right? You have that bacteria where its job is to destroy that bacteria so that it doesn't cause any problems. But it's also like your cat, right? For those of you who have cats know that one of the things your cats like to do is kill birds. But is killing the bird enough? Or does after it kill the bird, it wants to come in and plop it on your bed to show you what it did, right? So that's what these phagocytes do as well. Not only do they kill that bacteria, but then they take a little piece of that bacteria, shove it on its surface and go, hey, look what I just ate. I just ate this. This thing is running around. If this is bad, you might want to go do something about it. So not only do they break down these things, but they then provide that information to help to stimulate an immune response. I just killed this bad thing. I'm going to do something about that. And so they play an important role in that. Notice eosinophils are almost identical to neutrophils. They have a multi-lobed nucleus. They have granules. But what's the big difference you notice about the eosinophils compared to the neutrophils?
Color?
Color, absolutely. Again, color is one of those things we don't always pay attention to, but when we do, it's usually because it's color. It's usually because it's important. Here we have these massive, almost brick red, bright brick red vesicles or granules that are in them that contain the chemicals that they have inside of them to provide some protection. They also are phagolytic. However, eosinophils primarily play a role in two things. This is the actual color, absolutely. Yep, this is not the way they're stained. This is their actual color. And so when we look at these cells, blood in particular, typically not a lot of stain is used on them because they do have such distinct colors. There's still some stains that will put on it, but it doesn't change the coloration of this. This is actually what these cells look like. Eosinophils play a huge role in helping to deal with parasitic infections, especially parasitic worms. So if, for instance, you get some kind of lymphatic disorder like filarius or something along those lines, your eosinophils try to play an important role in helping to destroy, attack and destroy any of those parasites, especially parasitic worms. So they play a vital, vital role in our body's protection from those.
Phagocytes APCs.
I'm sorry?
Phagocytes APCs, antigen presenting.
Yes, antigen presenting cells, APCs, yes. Again, we won't talk about antigen presenting cells until we get to the immune response, although we will talk about antigens when we get to our blood typing later today. So yeah, so we will get to that, but you are absolutely correct. Neutrophils are one of the cells that can be antigen presenting cells. That is correct. And for those of you who don't understand what that means, don't worry about it. We'll get to it. All right. Basophils. Do basophils look like eosinophils and neutrophils? Do they have a multi-load nucleus?
Can't tell. It's too dark.
The truth is they do, but you can't tell by looking at them. Because as you look at this basophil, the basophil is filled with these massive, large, very dark granules that obscure everything else. And that's how it makes it easy to identify them. And it turns out these big, massive granules are just filled with histamine. Someone remind me what histamine does again?
Makes blood more leaky in the damage.
What do we call that process?
Inflammation.
Inflammation, absolutely. Histamine regulates inflammation. Basophils, along with mast cells, along with dendritic cells, are some of the cells that are in our body that contain histamine and help to regulate our immune response. All right? So what happens is when you're injured, as Oksana pointed out, they release their histamine and it causes that inflammation. You get redness, you get swelling, right? You get pain, right? And so redness, swelling, heat, and pain. Those are the four cardinal characteristics of inflammation. And it's caused by histamine. Histamine makes the blood vessels dilate so that they're more leaky and more blood comes to the area, causing that edema that we've talked about so that you know you're injured. It congests the area to protect it, and it also helps to facilitate healing, which is all awesome, except when that histamine is released inappropriately. When might it be released inappropriately?
Allergic reaction.
Right, in an allergic reaction, absolutely. Right, again, whether it is someone's cutting the grass, so the pollen or the other dust and irritants from the grass stimulate the release of histamine in your nasal cavity, causing inappropriate inflammation of your nasal cavity, causing congestion, increasing mucus production, causing your eyes to water, causing your nose to run, right? So what do you take to help to protect you from those grasses?
Antihistamine.
Antihistamines, absolutely. All right. There are two other leukocytes. They do not contain granules, and those are the monocytes and the lymphocytes. Notice the monocytes are by far the largest. When you look at the relative size of it compared to the red blood cells and those around it, it is by far the largest. And this is the one, as someone mentioned, is the big eater. When activated, it becomes the big eater. And what's the fancy word we use for that big eater? It's not just a phagocyte, but it's a...
Is it megakaryocyte?
No, remember megakaryocytes are the ones that make our platelets. Macrophage, there you go. Macrophage. Macrophages are the big eaters. When our monocyte is activated, it is our big eater. These are the slowest of our phagocytes to respond. So when you first injure yourself, a lot of them don't come out of into circulation. However, if you're dealing with a chronic infection, right, they're slow, but they're steady, and eventually the levels of them can get quite significant to help you deal with whatever that chronic infection is. And then lastly, notice again, when we look at the anatomy of it, it can be a little tricky to look at because notice it typically has, let's use a lighter color. It typically has a U-shaped nucleus. Notice not quite multi-lobed, but a U-shaped nucleus. And again, like I said, it is by far the largest of all of them, and it doesn't have granules the same way that the other ones do. Our lymphocytes, notice, have the least amount of cytoplasm. They have a huge, massive nucleus that almost completely fills the cell. Notice single-lobed, massive nucleus, very little cytoplasm, and there are no granules in the cytoplasm. And those are our lymphocytes. And our lymphocytes are the ones that are responsible for our immune response. As we will talk about later, those lymphocytes, they're one that eats the bad and the somewhat not so bad things. I'm not sure what you mean by the somewhat not so bad things. Phagocytes, again, eat things that are bad, but they can also attack things that, like for instance, if you have a liver transplant or things along those lines. I'm not sure what you mean by not too bad things. Yeah, so all of these, and again, when these function properly, these things do the right job. Where we have a problem is when there are inappropriate immune responses, right? Hypersensitivities or autoimmune disorders and things along those lines. And we'll definitely talk about that when we get to the next section. For right now, when we talk about our immune response, you may or may not have heard the term lymphocyte before, but lymphocytes actually go to one of two places in your body to mature. They can either mature in the bone marrow, or they can actually go to your thymus, which covers the superior part of your heart, and mature there. And anyone know what we call the lymphocytes that mature in the bone marrow?
B cells.
B cells. And those that go to the thymus to mature are our
T cells.
T cells, exactly. So when you hear people talk about B cells and T cells, they are talking about our two different types of lymphocytes. It's a great question. Why is one of those questions that is really hard to answer. Sometimes we're able to kind of tease out what the answer is, but as I've said many times, the engineer who designed all of this did not leave us her footnotes. So why is one of those things that we can guess at, but often we can't understand. And I honestly have no idea why the nucleus is multilobed. And again, it's not just that I don't know. I don't think anybody has an understanding of why it is that way. I don't have a good answer. They do produce a lot of proteins. That's what the vesicles are containing. So maybe there's an advantage to the differentiation of the DNA, make it easier to make the proteins that they need, but I don't know what that is. Notice lastly, and they've actually cheated for us here by giving us this right here. Notice what looks like a piece of dirt hanging out here on the slide, piece of chewing gum someone just left on the sidewalk. That's that cell piece that is the platelet. Remember, as we said, platelets are not true cells. They're just cell pieces. And we can see that here as well. So notice we see all of our formed elements here. In fact, I think I have it next. Excellent. Here is a picture I found many, many years ago. Hold on, let me do that. I like to believe it is real. It's like one of those things with the monkeys, slamming on the typewriters. You have enough monkeys slamming on enough typewriters, and eventually you'll get the complete works of Shakespeare. I would love to think if you have enough anatomists all looking at different microscopes and taking images at different times, eventually someone will get a picture like this, where all of the formed elements are present in a single image from a single slide. So like I said, if they faked it, then good on them, but I like this because this happens to show all of the formed elements. So let's go through them. Identify, whoops, too big, too small. Identify the formed element.
Erythrocyte.
Erythrocyte. Identify the formed element.
Neutrophil.
Neutrophil. Identify the formed element.
Monocyte.
Monocyte, excellent. Identify the formed element.
Leukocyte.
Lymphocyte. Lymphocyte.
Right, excellent. Identify the formed element.
Short again.
Neutrophil, another neutrophil. Remember, I said neutrophils were the most common. What's this one? Identify the formed element. Basophil, and identify the formed element.
Platelet.
Thrombocyte.
Thrombocyte. There you go. Don't write platelet on the exam. You write platelet on the exam, you will get it wrong.
Okay.
Well, you'll get partial credit, and it'll make
So-
you. So don't
so
make
The red one to the top right, it's eosinophil.
eosinophil. Yep, because of the red brick coloration. Yep. Excellent. Remember, I said the neutrophils are the most common. There's actually a great mnemonic that can help you to remember the order of most common to least common of our leukocytes, and that is never let monkeys eat bananas. Oops. So, what is the most common leukocyte?
Neutrophil.
What's the least common?
Basophil.
Basal.
Second
Basophil.
second most common.
Lymphocyte.
Or lymphocyte.
Second least common.
Eosinophil.
Oh, I like that one even better. Never let monsters eat babies. I'm gonna have to change that now. I love that. Never let monsters eat babies. I like that. Excellent. I always grew up on the never let monkeys eat bananas, but I like the monsters and babies way better. Oh, I'm totally stealing that, Alexa. I'm just warning you right now. And I'm not giving you credit for it either. I'm totally stealing that. All right. Questions on that? That's awesome. All right. Excellent. With that, we are done with our leukocytes for now. Like I said, we will talk about them more when we get to the next section when we talk about our immune response. Here we see that flow chart again, as we talked about, for hematopoiesis of our leukocytes. And again, remember, for all 5 of our leukocytes, you need to know both the primary and the secondary stem cells for them. But as I mentioned, I am not going to hold you responsible for all of the intermediate steps in between. So you don't need to know all the intermediate steps in between. You just, like I said, on that previous slide, right? So on any of these previous slides, let's go back here. On the exam, not only could I point at any of these formed elements and ask you to identify the formed element, tell me what that formed element does, but I also could ask you what its primary stem cell is and secondary stem cell. All right. Now, as we also talked about, right, it is going to be, oops, oh, I thought I had that on the slide. So I guess we talk about it more when we get to the immune response. Also, as I said, which direction it goes, whether it forms more eosinophils or whether it forms more lymphocytes, it's all going to be those local chemical signals in the red bone marrow that causes the differentiation. And as someone pointed out earlier, that local chemical signal are a big class of chemical signals called cytokines. So it is those cytokines that cause the differentiation to occur.
I have a question.
I have answer.
So for the erythrocytes and leukocytes, primary and secondary stem cells are hemocytoblasts and myeloid stem cell, right? Only for lymphocytes, the lymphoid stem cell, right?
Yes. So again, the primary is super easy because the primary is the same for everybody. So like I said, on the exam, if you see me ask what the primary stem cell of a formed element is, you don't even have to look at what the cell is because you know there's only one primary primary stem cell. For the red blood cells, for the platelets, and for 4 of the 5 white blood cells, myeloid stem cell is the secondary. The only one that's different is the lymphocyte. The lymphocyte is the only one that is formed by the lymphoid stem cell. That's why that lymphoid stem cell is a unipotent stem cell, because it's a stem cell that only becomes one thing.
Thank you.
Yep. All right. Excellent. Any other questions on that? All right, we're not done with our formed elements yet. We still need to talk about our thrombocytes. And then we have some other material that we need to cover, but this is a good stopping point. It looks like it's 1:10. Let's go ahead and take our first break at this point. Oops, I need that back. We will restart at 1:25, so we'll take a 15-minute break, and I will start the recording at that time. All right, so any other questions before we take our break? All right, I will see you guys in 15 minutes. All righty, let's dive in. We have talked about our erythrocytes. We have talked about our leukocytes. Any questions on those before we move forward? All right, excellent. Then let's talk about our thrombocytes. As we mentioned, a platelet is really just a cell fragment. It doesn't have a nucleus. It doesn't have an organelle. It's just basically a bundle of plasma membrane with a bunch of vesicles inside of it. Platelets, again, in a microliter of blood, more than the white blood cells, but obviously far, far less than the red blood cells. In that we have somewhere between 150 and 500,000 per microliter of blood. And as I keep saying, they're cell pieces. As we've mentioned, I'm not going to hold you responsible for the full path of this, but we do know that we have the hemocytoblast as our primary stem cell. We have the myeloid as the secondary stem cell. And then these cells become that cell that we already mentioned. And that yellow doesn't work well, so let's start a dark color. That megakaryocyte. A megakaryocyte is a cell that has undergone several mitotic divisions without completing cytokinesis. All right, what's that a fancy way of saying? If a cell undergoes mitosis but doesn't undergo cytokinesis, what can you tell me about that cell?
It didn't multiply.
True, but what did it do?
It copied its DNA.
It did more than copy its DNA. What else did it do?
It grew.
What's mitosis again, the process of?
Cell division.
Cell division.
It's not cell division. Mitotic phase is cell division, but mitosis itself is not cell division. What is mitosis?
Duplication of DNA and its organelles.
Right. Well, be careful that DNA doesn't have organelles, but you have the right idea.
And the
Mitosis is division of the nucleus. If you remember, prophase, metaphase, anaphase, telophase, as you go through those 4 stages, at the end of those 4 stages, you have what was 1 nucleus becoming 2 nuclei. Cytokinesis is the division of the cytoplasm, where we divide 1 cell's cytoplasm into 2 cells, and this is where we physically divide 1 cell into 2. These megakaryocytes undergo mitosis, but they don't undergo cytokinesis. So what ends up happening is 1 nucleus becomes 2, 2 nuclei become 4 nuclei. But how many cells do we have at the end of this process?
1.
Just 1, absolutely. And so that's what we have. We have this big, huge, multinucleated cell. And what's the advantage of having a lot of nucleus? A lot of nuclei?
More cells.
Well, true, we could get more cells, but what do nuclei contain again? DNA. And if you have more DNA, you have more instructions, and you have more instructions for making what?
Proteins.
Proteins. These platelets make contain massive amounts of proteins, massive amounts of vesicles. So what happens is megakaryocytes make a massive amount of protein, massively filling these vesicles. And then what happens, and here, let me cheat. I'm going to go ahead and remove some of this so I have some drawing space. Again, I'm going to have to work on my analogies, because normally my analogy, because we're sitting in the classroom, is I say that what we should do is go outside the building, go over to Auburn Boulevard, and stand on the sidewalk on Auburn Boulevard. And as you stand on the sidewalk on Auburn Boulevard, just for the heck of it, stick a leg out into the street. And what would happen if that occurred?
By a car?
Yeah, exactly. A car is going to come flying by because people drive about 90 miles an hour on Auburn Boulevard over by our school, and it would rip your leg off, right? You'd be left sitting there on the sidelines, and your leg would be going down the street. And that's exactly what happens. These megakaryocytes sit on top of blood vessels. So here's our capillary. And as we know, our capillaries have these intercellular clefts, these spaces in between. And so what happens is this big, huge megakaryocyte sits on top of the capillary, and it takes a little bit of its plasma membrane and it sticks it through that intracellular cleft into the cell. And then a big, huge red blood cell comes by and rips that little chunk of the cell off, and lo and behold, you have a platelet. And so that's basically what happens. This has all these processes that it sticks off into that capillary that get ripped off, and we get our production of our thrombocytes as a result of that. They fill themselves filled with vesicles, and these little chunks of plasma membrane with vesicles get ripped off and become the chunks of chewing gum on the side of the road that we see as those thrombocytes on the slide. All right, and it isn't much more sophisticated than that. What do our platelets do?
Blood clotting.
Right, they play an important role in the maintenance of our blood volume by forming clotting to occur, platelet plugs that occur, and we'll talk a little bit about that process. In general, they're one of the important steps in a process called hemostasis. Hemostasis is a sequence of responses that, again, play a role in helping us from maintaining our blood volume, stopping the loss of blood from bleeding. This is actually a three-step process that our platelets play a role in. Because the testing is in just a few days, we're not going to go into too much detail. Your book does a really nice, elaborate job about talking about some of these processes. We're not going to go into it in that depth, but we do need to know the three phases and the three steps involved in this. So we will briefly talk about these things, but not in too much depth, just to get in a general concept. So there you go. Here we have it all put back together again, the entire process of making the formed elements, the hematopoiesis, where we produce all the formed elements, whether it is coming over this way to produce our red blood cells, or whether it's producing the megakaryocytes that give us our platelets, and so on and so forth. And to truly beat the dead horse, as we mentioned, this whole process is driven by hormones and chemical signals. And we've talked about some examples of those. Erythropoietin, again, which produced in the kidneys, targets the bone marrow, leads to an increase in red blood cell production and increases the rate of maturation. Thrombopoietin is actually produced by the liver and plays an important role in stimulating our platelet formation, getting more thrombocytes. And as we also mentioned, it is those local chemical signals, those cytokines within the bone marrow that cause the differentiation of the different types of white blood cells. So for instance, some examples, cytokines, remember, it's going to be important to remember, this is a very large class of chemical signals.
Can't see that. Or I
Correct, you cannot see that. There we go, how's that? Better? Large class of chemical signals are our cytokines that include things like colony stimulating factors, interleukins, and all these types of things. And again, you'll get to some of those in micro. We'll get to some of those when we get to it in the immune response. But like I said, there are these local signals in the bone marrow. And if we cheat and actually go back to the previous picture, we can actually see some examples of this. So notice erythropoietin causes the differentiation of these cells, causes them to speed up their production. Colony stimulating factors G and M, and multi-CSF. All of these are different types of things that lead to the differentiation of different types of cells that are occurring. And again, you don't have to memorize these names of the different colony stimulating factors or anything like that, Lon. I just want you to remember that this differentiation is driven by hormones and chemical signals. All right, there. And those are two names that you should know: erythropoietin and thrombopoietin. Excellent. All right, so the last little thing we need to do for our blood is, like I said, this process of hemostasis. Hemostasis is the three-step process by which we try to maintain our blood volume when injured. Step one in this process is what is known as the vascular phase, or what is also known as the vascular spasm. Step two in this process is the platelet phase, where we form our platelet plug. And that leads to the coagulation phase, which causes the clotting of the blood, the formation of the scab, and ultimately sets the framework by which the healing process can begin. Your book's got a pretty picture that goes along with some of these, but let's talk about them, like I said, in a little bit of detail, talking first about our vascular phase. Vascular phase is triggered when the blood vessel is damaged. When the blood vessel is damaged, that blood vessel is going to do two things. Obviously, it's going to stimulate pain receptors, making you aware of the fact that you're injured. But there are also chemicals that are released from the damaged tissue. So again, what we are getting here is both a neural from the pain receptors and a chemical control. And what this neural input and chemical input causes the blood vessel to do is it causes the smooth muscle of the blood vessel to contract. That is why this is also referred to as the vascular spasm. And so what happens is a blood vessel constricts. And as we know, when a blood vessel constricts, we reduce the diameter. And when we reduce the diameter, what do we do to the blood flow?
Reduce it.
We reduce the blood flow, absolutely. Right? So this reduces the blood flow. This contraction of the smooth muscle doesn't last a long period of time, only about 30 minutes. So what that means is if I get my arm cut off by a machete, I'm okay for the next 30 minutes because my blood vessels are going to contract, stopping the blood loss, and I can finish my sandwich before I get to the hospital, right?
No.
No, obviously for the large blood vessels, this is not going to stop the blood flow from that high pressure. So as you see, this works best in the smallest blood vessels, paper cuts and superficial wounds, things along those lines. But it can play a role in helping to limit the blood loss from those smaller blood vessels in those cases. It's not going to be nearly sufficient to do anything on its own or for the large ones, but it can play an important role for limiting blood loss in smaller blood vessels. But even that doesn't last forever. And so luckily we have other features of our hemostasis that is going to help us to maintain our blood volume. And the next one is a process that we talked about way back in 430, and that is our platelet phase, or our platelet plug formation. Right? As we mentioned, our platelets contain two types of granules, lots of different types of proteins. But we didn't really talk about what they were, so let's do that a little bit. The first are alpha granules. They contain the clotting factors. They're the ones that are going to play the big role in helping us to stop our blood loss. But they also have some growth factors. Growth factors are hormone-like chemical signals that are going to help to cause the repair, and they play an important role in producing, let's say it that way, the feedback mechanism of our platelet plug formation. And can someone from 430 remind me again what type of feedback mechanism blood platelet plug formation is again? If only I knew what the different types of feedback mechanisms were in maintaining
Negative.
homeostasis. Negative and positive, absolutely. Which one's the more common one?
Negative.
Negative.
Negative feedback. And which one is platelet plug formation?
Positive.
Positive. This is one of the
Positive.
3 classic examples we have of a positive feedback mechanism. What were the other examples of positive feedback mechanism? Give me at least one of them.
Childbirth.
Childbirth, right? Labor, absolutely, childbirth. Something else we'll talk about in this class as well. There are others, but those are probably the 2 most classic that most people know about. This is what leads to that positive feedback mechanism, right? It's here, these alpha granules. Now, repair requires a lot of other resources. So those platelets also have what are known as dense granules that have things like ADP and ATP, some energy molecules. Calcium's there. Why might we need calcium? What's calcium make cells do?
Wonky things.
Wonky things, absolutely. And healing is definitely a wonky thing. Other enzymes and chemicals that are going to help to facilitate this process. So here's how this works. Again, I think we talked about this in 430, but we can go ahead and talk about it here. Let's go ahead and draw a blood vessel. Here is our blood vessel, and we will put a cut in our blood vessel. When we put that cut in the blood vessel, the damaged tissue, and so let's do that, and let's do that. Our damaged tissue and the surrounding cells, the damaged blood vessels, will release a chemical signal. And what happens is that chemical signal causes a platelet to come and attach itself to that damaged tissue. So a platelet will come and attach itself to that damaged tissue. When it attaches to that damaged tissue, it undergoes this chemical reaction that causes it to release most of the contents of those granules. It releases the energy molecules and the enzymes, the clotting factors, and that platelet-derived growth factor. And that platelet-derived growth factor is a chemical signal, and that chemical signal will attract more platelets. So now more platelets come to the area, and those platelets attach. When they attach, they undergo that chemical change, and they release even more of that chemical signal. Remember, that's a key with a positive feedback mechanism. With a positive feedback mechanism, each time we are enhancing the stimulus, right? Negative was easy. If I want to balance something, my pen I think shows up better. My pen starts to fall to the right, I can cancel that or negate it by bringing it back to the left, right? I've maintained balance. In this case, I'm tilting it to the right, and I tilt it even more. I'm enhancing the disturbance, releasing even more chemical signals. But as I release more chemical signals, more platelets come and attach. And as more platelets come and attach, they release even more of the chemical signal. And notice it keeps enhancing, and it keeps enhancing, getting bigger and bigger and bigger until, no, I didn't want that to be red. Until ultimately, we have so many platelets coming and attaching to the area that we have formed a platelet plug. And when we form that platelet plug, that helps to stop blood loss. Now again, is this going to help stop blood loss in my brachial artery if that's what I've cut?
No.
No, obviously, again, this is going to be most efficient in the smallest. And the other thing to remember is platelets are just pieces of plasma membrane. So basically, I've just got a bunch of chunks of plasma membrane that are going to form here and help to form this stop gate that is going to help to stop the blood loss. But little pieces of plasma membrane by themselves are not going to be enough to stop the complete loss of all of our blood. And that's where these clotting factors come in. The clotting factors are going to play an important role in the third phase of hemostasis, which is that blood clot formation. All right. But in this case, we have that positive feedback mechanism that leads to the formation of our platelet plug, right? We just did this. Platelets attach to the damaged epithelium. They undergo a release reaction, which causes them to swell, causes them to release their contents. And those chemical signals attract more platelets to the area, forming a seal for that minor damage. And I think I have a pretty picture of this. I thought I had a pretty picture of this. Guess not. All right. Questions on that? All right. But if we really want to seal this, great question. I know there are medications that can be taken that decrease the rate of clotting. I'm not sure if we have ones that would increase, because that wouldn't necessarily, as a medicine, at least as a systemic medicine we took, I don't think that would necessarily be a good thing, because we don't want to increase the likelihood of platelet plugs forming, because then that can lead to clots forming inside the blood vessels that could then break off and become and cause embolisms or things along those lines. So I know we have medicines that do. There might be something topical that can be put on. I'm not familiar with that, but there may be something like that. I think more we rely on pressure and things along those lines.
I mean to say count and not that really.
That's all right. I understand. Don't worry. I saved the chats forever, and I'll be sharing that with
Oh
them.
my God.
I'm just kidding. All right. Excellent. All right.
Apparently, there are actually drugs that are anti-fibronil. I can't say it. Fibronil drugs.
Yeah. Well, like I said, heparin and things like that will deter the formation of the clot. Right? Heparin is an example of one of those. But that's like I said, if you take that, for instance, because you want to worry about, clots that are forming inside of the blood vessels, you want to worry about embolisms, or you want to worry about somebody who's had a heart attack or something like that, then that's a good thing for that. But then if they cut themselves, right, then they have a hard time stopping the blood loss from that because they're taking those. So like I said, I know we can do the opposite. I don't know about the other. I guess if we had low platelets, thrombopoietin, remember, is a hormone released by the liver that can increase platelet formation. And I guess if you had abnormally low platelets, there might be a man-made version of thrombopoietin, or maybe like pig thrombopoietin or something like that, sheep, cow, horse thrombopoietin or something like that we take that would be able to increase that. Not my area of expertise, so I don't know that for certain. Interesting. And did they give you some medicine for that low thrombocyte count?
Yeah, steroids.
Oh, interesting. Oh, anti-fibrolytic drugs. Oh, interesting. Again, not my area of expertise, so I'm a brain guy. That's really cool. So yeah, apparently there are some things. So it looks like it discourages the breaking down of the clots, but it doesn't necessarily enhance platelet plug formation. But I'm sure there's something like that. Again, part of the whole reason that we learn how the body works is so that we can learn how to manipulate it to our advantage. So that doesn't surprise me in the least. All right, let's again briefly, I will tell you, your book does a beautiful job of talking about the coagulation phase and the steps. It has one of those massive, and there's the special word, cascade events. What do I mean when I say cascade events?
Step by step.
Yeah, it's like those dominoes. You hit one domino and it hits the next, and it hits the next, and then it hits two, and they go off on the different paths, and then all these magical things happen, right? That's what it is. And your book actually does a really nice job describing these processes. We're not going to go that much in depth with it, but not surprisingly, there's a lot of stuff that might help with hemophiliacs. That is definitely true. People have problems with coagulation. Absolutely. I hadn't thought of it from that standpoint, but that does make sense. Not surprisingly, again, all sorts of things are necessary for the clotting. Like I said, vitamin K, calcium, liver enzymes, those chemicals released by the platelets and by the dead and damaged tissue, all of those things occur. And like I said, we have this massive cascade of reactions that takes place. I think I actually have the pretty picture here for it. Here's the pretty picture. So again, your book does a nice job of describing this. I'm not going to make you know that enzyme X2 is converted into enzyme X2A activated, which then converts X1 into X1A and so forth and so on. I'm not going to make you need learn this. If you have trouble sleeping one night, I encourage you to go and take a look at this pathway and read it, because it'll put you out in no time at all. But the real key that I want you to get from this, is that there are basically two main pathways, as you see. And this illustration does do a nice job of emphasizing that. The first is the extrinsic pathway. The extrinsic pathway is primarily caused by damage to the tissue itself that has been damaged. Right, so again, the surrounding tissue, you get a cut to your skin or something like that, that surrounding tissue, the thing's basically outside the blood vessel. And that's really the key with the extrinsic. Tissues outside the blood vessel get exposed to the blood and cause this process to occur. Whereas the intrinsic pathway involves things within the blood vessel. So this is either damage to the endothelium of the blood vessel. This is the chemicals that are produced and released by the platelets, all of these things that are inside the blood vessel. And that's really the difference. Things outside the blood vessel and inside the blood vessel both have the ability to have an effect. But the real key, as we've talked about, is this effect. What we care about, what's going on, is really this right here. This is really the take-home message. Both the intrinsic and extrinsic pathways essentially want to do the same thing. We need to convert that water-soluble fiber, that is fibrinogen, into a water-insoluble fibrin. That water-insoluble fibrin. Because when we do that, what happens is we form this big, huge, massive fishing net, right? Back in ancient times, right, when people would fish in the ocean, they would use these big, massive fishing nets. And they used it for tuna fishing, for instance, all the time, right? Because everybody loved their chicken of the sea, their tuna, and Charlie needed to be on the shelves. So you'd have these fishermen with these big, huge, massive nets catching that tuna to be able to serve it up on your plate. And what was the problem with those fishermen? Anyone old like me remember back in the days when there was a big, huge issue with tuna? What was the big, huge issue with tuna? Anyone remember? Maybe your grandparents told you about it.
You catch everything else in the net.
Yeah, and primarily what was the thing that they were worrying about catching?
Dolphins.
Dolphins, there you go. Can't kill the dolphins, right? Absolutely not. So it would catch everything. Right? Now, when we're talking about dolphins, that's a bad thing. But when we're talking about pathogens, red blood cells, white blood cells, and all this, forming this massive net that captures and clusters everything together, forms this big, huge protective clot over the top of the damage. And then notice also, these fibrin fibers that are in there, they actually tighten. And as they tighten, they draw water out of them. So these fibers actually dehydrate. And as these fibers dehydrate, that's what forms the scab. So that scab that forms on the top of your injury is caused by the dehydration of this fibrin that has been brought out of solution by this elaborate cascade process that is our coagulation phase. So like I said, I don't care if you know anything about the process or the phase, but know that this is what coagulation is. It's the pulling of that water-soluble fibrinogen out of solution to form this massive fishing net to catch and stop everything so you stop losing blood, and then it forms the framework of the scab to allow the healing to occur underneath.
Hydrates fibrin.
Yes. So again, fibrinogen, and so here, we'll cheat and go back, right? Water-soluble fibrinogen becomes the water-insoluble fibrin. That's really the key to coagulation, because then you form this big, huge fishing net that catches not just tunas, but dolphins, mermaids, right? Everything in the sea gets caught into that net, and it stops the loss of blood. All righty. Questions on that? Excellent. All righty. The last thing we need to talk about for today is to talk about blood typing. And again, we're going to do this even though you had like I said, a lab simulator that talked about this. So hopefully these are concepts that make some sense to you. I think it is an excellent lead-in to the things that we are going to talk about next, where we move on to our immune response. Hold on, there's a quick question. Ultimately, the goal is to not just stop the loss of blood, but also stop the loss of water as well, right? That's one of the reasons why these fibers tighten and become that hardened scab, to form more of a water, moisture losing surface. But yes, I mean, as anybody who's had an injury knows, can they seep interstitial fluid from them? Sure, absolutely. But the goal is to not just lose.
There is still a way.
I'm sorry?
Oh, I just want to ask, but there is still a way for the bad stuff to go out, like pathogens.
No, but remember that so we get the injury, we get the scab, but then we get the inflammation in that area. And that inflammation attracts white blood cells. The chemicals that the platelets release are going to attract white blood cells. So like I said, we will definitely, in the next class, when we talk about our immune response, we will talk more about some of these concepts.
Okay.
Okay. So infections and scabs, like I said, let's talk more about those things when we get to the next part of our immune response. Yeah, we'll talk about that more when we get to our immune response. All righty. The last concept, and again, this is a good feed into what we're going to be talking about in the next, is one of the most important things we have to do for the survival of ourselves is to be able to have our body be able to recognize the difference between things that are us and things that are not us, or between self and non-self. And the way we do that is twofold. The way we identify ourselves with special molecules on the surface of our cells. And these special molecules on the surface of our cells are known as antigens. Antigens are primarily proteins, but can also be carbohydrates or even lipids. Or they may be glycoproteins or all sorts of different combinations of these things. And so if we have a cell in our body, like this cell right here, it can have any number of these special markers on their surface. Now, right now, we're talking about blood cells. So our red blood cells in particular are somewhat unique, and they only have a few of these proteins on their outer surface. In fact, they only have around 30 or so antigens on their surface. And those antigens on the surface are the markers that help us to tell the difference from people that are us and people that are not us, things that are us and things that aren't us, cells that are us and cells that are not us. Now, right now, there are 28 of us here sitting here talking on Zoom in this classroom. Do you think that any two of the 30 of us in this class have the exact same 30 antigens on the surface of their blood cells?
No.
Maybe
No.
not.
Absolutely not, right? Absolutely, absolutely not. Does that mean that nobody in this class could give blood to somebody else inside of this class?
No.
No.
No, as it turns out, there probably is. Even a group this small, I'm fairly certain there's probably a handful of people that would be able to, one person could give their blood to another. The reason for that is while our red blood cell has 30-some antigens on their surface, there are only 2 that cause a severe reaction. And you may not have thought of it in these terms, but I know most of you know this. Because most of you, hopefully all of you, but I'm guessing at least most of you know your quote unquote blood type. How many people here know their blood type?
Yep.
I know.
A couple people. Give me some examples. What's your blood type?
O positive.
Excellent. I see a B negative. I see an O positive. I see an ME, an A negative, an A positive. Excellent, right? Notice we have these blood types, and when talking about these blood types, there's typically 2 components that we talk about when we give our blood type. We give some kind of letter, and that letter is usually some combination of, as we see here, A's, B's, and O's. In fact, this particular antigen is called the ABO antigen because of that. And then usually there's some kind of either plus or minus sign to it. And that particular antigen, anybody know what that is?
RH antigen.
Excellent. It is the RH factor. Excellent.
Rhesus factor.
Rhesus factor, exactly, named after the rhesus monkey where this was first studied. Absolutely. So this RH factor. These are the 2 antigens that are on the surface that cause a severe reaction. So those are the ones that we care about. What we care about when we want to give blood from one person to another, right? It's pretty easy because we only have to worry about these factors. On the other hand, if we were dealing with something like a liver cell, right? With a liver cells have over 200 antigens on them. And far more of those cause a severe reaction. So while I guarantee there's at least a couple people in this class who could share blood, or at least one person could give it to the other, are there likely to be 2 people in this class who one person could give half their liver to the other?
Nope.
Right. Unless I have siblings taking this class, then that's probably not the case. And even with siblings, right? Obviously, identical would be easy, but just regular siblings, even then, it's not quite likely. Right? So that's the key. We have these molecules on the surface. And again, for simplicity, we'll draw them like things like circles. We'll draw them as things like triangles or things along those lines on the surface of them. They're these antigens that we need to be able to show that this cell is me or this cell is not. All right? Are we comfortable with that? Excellent. Silence means yes. So the other side of this is once we have this stuff on our cells, we have to be able to recognize cells that are not us. So say my cells have circles and triangles on the surface, but there's some foreign cell, and that foreign cell has squares on the surface. Now, obviously, are any of these things really square shape or circle shape or triangle shape? No, of course not. I'm just doing this for simplicity. But my cells have circles and triangles. So what I need to do is not just have the right things on my cells, but I have to be able to recognize things that are not me. And I do that with antibodies. Antibodies are proteins that are used to signal, used to identify and signal things that are not us. So in my blood, what I need is an antibody. These antibodies, actually I should be even more specific than this. These are Y-shaped proteins with 2 binding sites. So in my blood, so it's my blood, so I'll go ahead back and make it red again. I have, whoops, that's not red. That's red. Floating around in my blood plasma, I have these Y-shaped proteins. And this the job of this Y-shaped protein is to recognize this abnormal cell. And the way it does that is by having a specific binding site on it. And notice in this case, this specific binding site would be square-shaped. And that square-shaped binding site would allow that antibody to be able to bind onto that foreign cell. And it's kind of like planting a flag in that cell, saying, hey, everybody look over here, this thing doesn't belong. And it basically is a flag that gets waved, and that attracts our phagocytes, or that causes other things to help to destroy and eliminate, or at least neutralize this abnormal cell. So there are 2 key things we need to tell the difference between us and something else. And those 2 key things are antigens on the surface of our cells and antibodies in the blood plasma. Well, let's say it this way, free floating in the blood plasma. All right?
I have a question about that.
I have an answer.
So our antibodies have the receptors for cells that aren't ours?
Correct.
Okay. Okay, that was it.
Yep. Let's see how this works. Let's cheat and go to our whiteboard. Clear that. And draw 2 big fat lines. Line 1. Line 2. Now, we are going to actually, now this one. I also need cells. There's a cell. Cell. There's a cell. And there's a cell. Now, we are going to focus first on the ABO antigen. With the ABO antigen, there are, let me make that a little smaller. With the ABO antigen, you have some combination of 2 possible antigens. And those 2 possible antigens happen to be A and B. So let's say, for argument's sake, because it kind of looks like it, our A is our triangle. So here we have a red blood cell that has triangle-shaped A antigens on its surface. What do you think we call the blood type of a cell that has A antigens on its surface?
It's the A blood type.
Yeah, blood type A. So this person is blood type A. Excellent. What if instead the person has just B antigens, and we'll make those round. B antigen, B, 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 B. Excellent. So in this case, we have a cell.
That's B type.
Right, there you go, exactly. So this would be our B blood type because it has the B shape antigen on its surface. So only B antigen on its surface. Oops. So there we go. And we would say that there. And we should put that back on the other one as well. So let's sneak and put an only over here. However, it is possible to have cells that actually have both A and B antigens on its surface. And what would we call that person who has both triangles and circles on their surface?
Type AB.
Type AB, excellent. Let me finish with my amazing drawings here. There, excellent. This person we would say is type AB blood, but also, there. It is actually possible to have neither A nor B antigens on its surface. So it doesn't have circles or triangles on its surface. And what would we call this person's blood type?
Type O.
O
That
type.
person is type O. O type blood. Perfect. All right. That part makes sense?
Yes.
Excellent. So now let's go back and talk about the flip side of this. This person who has the type A blood, they need to protect themselves from cells that have the protein that is not them. What is the protein that is not them?
B.
The B.
So what they need is oops, that's going to be way too big. There we go. So in the blood plasma, they need an antibody that can recognize B antigens. And we have a fancy name for that antibody, and the fancy name for that antibody is anti-B antibody. So if we're going to draw this for their blood, they're going to need to have that Y-shape antibody. And based on this simple illustration we've been doing, what should the shape of their binding sites be?
There are half.
Round.
Yeah, circles. So they need the circles. Exactly. So that anti-B antibody would allow them to recognize things that are not them. All right. This person over here in the blood plasma, they need an antibody that recognizes what?
Circles. A antigens.
Excellent. A antigens. And what do you think the fancy name for that antibody is?
Anti-A antibody.
Anti-A antibody. Excellent. It's going to be a Y-shaped protein. And what shape is its binding sites going to be?
Triangular.
Triangles. Oops. Okay, that one's a little weird shape, but you get the idea. So what's going on here with our AB fella?
It has no antibodies.
Exactly. Notice both A and B are it. So it doesn't need to recognize a foreign antibody or antigen. So because of that, there are no antibodies in their blood, in their plasma. Now, again, does that mean they have no antibodies at all? No, we're just talking about there's no, so let's even be more specific. No anti-A or anti-B antibodies in their plasma. Because triangles are them, circles are them, they don't need to worry about recognizing either. But what about our O friend here?
It has all the antibodies.
Right, here, neither A nor B is us in this case. So we have to recognize both A and B antigens. So it needs both anti-A and anti-B. All right. And so let's go ahead and draw that. Oops, I need to move this out of my way. And we'll just draw them this way because it'll be easier. There's one Y, there's our second Y, and one is going to be triangles, and one is going to be circles. Excellent. Oh, and because I'm anal, I need to release that and make that that. Excellent. So notice now for our ABO blood type, we have identified, B, there we go. We have identified all of the 4 possible combinations of both antigens and antibodies that a person could have. Every single person in this class falls into one of these one of these 4 categories. Okay? Now, Ryan's off to a good start when we talk about universal recipients, right? We'll get to that in a little bit more detail in just a second, but you have the right idea. As we talked about, we want to have the ability for one person to be able to give blood to another. So for instance, in this particular case, and I'm just going to go ahead and write this just for this one here, for this person who has A blood, what blood could they receive?
A and O.
Excellent, right? Notice you can always get your own type. If you were type A, you can always get type A. But notice the nice thing about O-type blood. O-type blood doesn't have triangles or circles on the surface. So it doesn't have anything on there that your antibodies could attach onto and say, hey, this doesn't belong. So O blood could be given to A. In fact, O's blood can be given to who?
Anyone?
Everyone on this list. Everyone on this list can get O's blood, right? Now, who can A give blood to?
A or AB.
Okay, it could give to A or AB, right? Because it can't give to B. B's got the triangle antibodies. Can't give to O. O's got the triangle antibodies. But A, again, you can always give and receive from your own type. B, notice, doesn't have either antibodies. So they are able to receive all types of blood. Okay? Now, before we finish with the rest of these, I want to go back and add one more component to this. Okay? Any questions on the ABO before we move forward? Are you guys still there?
Yeah.
Okay.
Yes.
I got a warning on my Zoom saying that my internet was unstable. Am
You did
I
break-
breaking up, or can you guys hear me okay?
You were, but you're good now.
Okay, excellent. Okay, perfect. Good. The sun went away, so hopefully that's good. All right, excellent. So when we talked about people's blood types, they said A, they said B, they said O. I don't think anybody said AB. Is there anybody here who is AB? No one who's willing to admit it. Okay, excellent. But as we also said, people didn't just say A or B, they said positive or negative. That positive or negative refers to the Rh factor, as we mentioned.
May I ask you before you go forward?
Yes.
Why A can be given to AB? There are still anti-B antibodies.
True, there are these antibodies, but these proteins aren't as numerous in their blood. So when you give blood, typically you're giving the blood cells to that person. So you don't have to worry about this person's antibodies. You have to worry about your antibodies. AB doesn't have any antibodies there because it's really not even the antibodies.
Oh.
It's also that if you have antibodies, you can make more antibodies. So if AB blood is given to A, A's antibody will bind to this, and that will stimulate a response, and you'll get a lot more anti-B antibodies that will cause problems. AB blood doesn't have any antibodies, so they don't have to worry.
Okay. Okay, got it.
Okay?
Thank you. Mhm.
Yep. All right. So, excellent. All right, so hold on. I want to shoot and receive blood. Good. Okay, or good. All right, so then I can erase this, and now I can switch my text to green, and we can talk about, oh, I didn't know I could do that. That's cool. Did your image change of this at all?
Yeah, it shrunk.
Oh, it shrunk. Interesting.
Like,
For me, it got bigger. Now is it bigger or smaller?
Bigger.
Interesting. Okay, cool. All right, well, I didn't know I could do that, but that's fun. All right, so now we're going to talk about the RH factor. RH factor is a second separate antigen that comes in two forms. You either have it or you don't. That's what basically positive and negative means. If, for instance, you have it, and let's go ahead and draw this as a green square on the surface. Then that means that you have the RH factor. And if you have the RH factor, how do we typically identify those individuals?
Positive.
Positive.
Right. They are positive. So this person is a positive. Okay. However, if you do not have the RH factor, how do we identify those people?
Negative.
Negative, excellent. So they're going to be negative. And minus. And not only are you going to be minus, but if you don't have the RH factor on your blood, what do you need to be able to recognize this?
Anti-RH antibody.
Exactly. You're going to need, so not only do you not have the RH factor antigen, but you also need the anti-RH antibody. And in this case, that's going to be a Y-shaped structure with what shape binding site?
Square.
Square.
Professor, I had a quick question.
Yes.
Was it, so I know you call it the RH factor. Would you mark it wrong if we called it the RH antigen, like on an exam?
Yeah. RH factor antigen or RH antigen, I would be fine with that. Yes.
Okay.
Okay. Excellent. This person here has the RH antigen on their surface. So how would we identify this person?
AB positive.
Excellent. This person here is going to be AB positive. And if you have it's so hard to switch back and forth colors. If you have the RH antigen on the surface, then that means no antibody. Whereas if you do not have the RH antigen on the surface, what does that mean?
You have the antibody.
You must have the anti-RH antibody. Excellent. And so for this one, we need one last Y with a square shaped binding site. All right. And of course, this person, what would this person's blood type be considered?
Oh, negative.
Red cell donor.
Right.
No, negative.
O negative, and you are absolutely right. As was just mentioned, this person is considered the universal donor because much like the top of my head, there is absolutely nothing on this red blood cell, right? And as such, it can be given to anybody. When the apocalypse comes, which is happening more, looking more and more like it's going to be happening quickly, if you know anybody with O negative blood, grab them and lock them in your basement because they're going to be a valuable resource to you, right? Because they're like bright and shiny, baby butt, clean and clear red blood cells that can be given to anybody. On the
My
other hand,
husband's O negative.
I'm sorry?
My husband's O negative.
Excellent. Hold on to him. At least until 2020 is over. Once 2020 is over, then feel free to dump him. But for the rest of this year, make sure you hold on to him. Right? Doesn't look like we have any AB positives, but if you were AB positive, notice you don't have any antigens at all, right? And so what do we call that person?
Universal receiver.
Universal receiver. If you've got AB positive blood, you can basically see a pool of blood sitting on the street, pick that up, throw it in your arm, and be perfectly fine. Okay, don't do that.
For hepatitis and other bloodborne
Yeah,
diseases.
okay, sure. Okay, so there might be some bad things, but your blood's not going to collect it. So that's going to be the one miss. All right.
So all
Go ahead.
all type blood can be given if it's positive. All of them, only who's also positive.
Correct. And that was going to be what I was going to come back to. Let's go back to what we did here and put a little line through this and be more specific this time. So now that this person is, we know this person is A positive, let's be more specific about what they are able to receive. So blood, they can receive. If you are A positive, what blood can you receive?
A and O, positive and negative.
Excellent. You can get A positive. Remember, you can always get your own type. But you're right. You can also get A negative. Right? You can get O positive. And everyone can get O negative. So when you're figuring out what people can receive, the two easiest to figure out is that everybody can get O negative and you can always get your same type. So A positive can always get A positive and everyone can get O negative. So then what you have to do is you have to look at your antibodies. This person's only antibody is anti-B. So the only thing they can't receive is something that has B. So they can't get B positive, they can't get B negative, they can't get AB positive, they can't get AB negative. Who can they give blood to?
A positive or AB positive.
Right, A positive or AB positive. Notice someone who is AB negative would have the Rh antibody. Only AB positive is the universal receiver. AB negative is not a universal receiver because they would have the anti-Rh antibody. And as we said, O negative is the universal donor. So notice, again, I don't remember who said it. Somebody mentioned when we were talking about O before being the Orion, about the universal recipient. Really, the universal recipient, the universal receiver is not just AB, but AB positive. Our universal donor is not O, it's O negative because of those two antigens on the surface. All right? So hopefully this makes some semblance of sense. Questions on this?
What is the problem sometimes when mother's blood have different time from babies, like in the positive and negative? I heard about it, but
excellent
how it
question.
works.
Absolutely. So let's say, for instance, this is your blood type B negative. If your blood type is B negative, notice you have that Rh antibody. But your husband could have positive blood. And so what that means is when your genes combine, you put those puzzle pieces together to make a baby, that baby inside your tummy could have a positive Rh factor blood, and you have anti-Rh antibodies. Now, typically with the first baby, because these antibodies typically don't cross the plasma membrane, it isn't much of a concern. But as many of you know, giving birth is a messy process. And during that messy process, baby's blood often comes in contact with mom's blood, and mom's blood then sees that Rh factor in baby's blood and goes, well, I was exposed to this, and it builds up a big, huge, massive defense. So that when you have a second baby, and that second baby is also Rh positive, you have enough of a defense in your Rh antibodies that it can actually cross the placenta and it can attack the blood vessels, the blood cells of the baby. So that's that Rh incompatibility that can be an issue. And so what they need to do is suppress that immune response, typically with some kind of medicine like RhoGAM or something along those lines. But that's what it is, because it's only an issue for moms who have a negative blood type because they have those Rh antibodies. So typically if you're negative and your husband is positive, then they will do a blood test on the baby to see, or they'll just give you even RhoGAM just prophylactically just to make sure.
Okay, thank you.
Yep.
It's only relevant for like the subsequent babies, right? The first
It
one.
is typically it is more relevant for the subsequent babies, absolutely. In rare instances, again, depending on what's happened to mom, especially if mom's a nurse or something who may have come in contact with positive blood in some other way, then it is possible that it could have built up a defense to it already. So in rare instances, it is possible to attack the first baby, but usually it's much more of a concern for the second. All right. All right, we've done this here. We can do the whole thing again here. Like I said, we need to be able to recognize our cells. We use those tags that are the antigens. Mostly proteins, but they can be combinations of also proteins, carbohydrates, and lipids. And they form those tags on the surface that allow us to identify them. But we also need to be able to recognize things that aren't us, and those are those free-floating antibodies. If I remember correctly, it's either your Labster or, I think it was the Labster, where there are some antiquated terms, agglutinins and agglutins. Those are just more antiquated terms that are used for antigen and antibody, but they're acceptable terms and they're fun to say if you want to use that. Like I said, those antigens on the surface of the red blood cells, there's about 30 of them on there, but there's only 2 that trigger a severe response. And here we see that combo like we did, type A. And ironically, you notice your book's got it the opposite of us. The A's are circles, the B's are triangles. But again, neither of those are accurate. Those are just simplifications of them. And then, like we said, the antibodies are used to identify them. So if you have type A blood, you need the anti-B antibodies. If you have type B blood, you need the anti-A. If you're AB, you have neither. And if you're O, you have both. And like we talked about, that affects what type of blood each person can get. All right. Now, these blood types are not randomly distributed throughout the population. Here we see the typical population breakdown of the different blood types. And again, notice this also isn't taking the Rh factor into account on that as well. What we're left with is, while most people in here knew their blood type, or at least mentioned their blood type, there may be some people in this class or some people elsewhere who don't know their blood types. And so the way we learn that, and the way is to take advantage of this defense mechanism of the agglutination. This is why we don't put bad blood into our bodies. As I mentioned, and let's cheat and do a little drawing here. Our antibodies are these Y-shaped proteins that can bind to a red blood cell that doesn't belong to us. But as I mentioned, they have 2 binding sites. So that means one antibody can bind to 2 red blood cells. And then maybe another antibody binds to a third, and another antibody binds to a fourth. And notice suddenly what we get is this clumping together of the blood cells. And if you start clumping together the blood cells, is that going to make it easy for these things to move through your blood supply? And what's going to happen
Not
when a
at
big
all.
clump of red blood cells get to your capillaries that are only one blood cell thick? Are they going to be able to get through those?
No.
No. So this agglutination, this clumping of the blood, is a very, very bad thing that we want to avoid. However, we can take advantage of it to understand using this clumping to see how our blood types work. What we can do is we can take a sample of blood. So if you don't know what your blood type is, we can take a sample of your blood and in this case, put it in 3 different wells. So here is your blood with 3 different samples of your blood. And into this first one, we add some anti-A antibodies. To the second one, we add some anti-B antibodies. And to the third, we add some anti-RH antibodies. All right? Now, let's think about what happens. One of 2 things is going to happen in this. If this cell has the A antigen on it, what's that antibody going to do?
Gonna bind
Bind to
agglutinate.
it and it will clump it together. And notice if it has the antigen, if the antigen is present, it will clump the blood together. But if it doesn't have the antigen, does it affect the, can it affect the blood?
No.
Right? And so if there's no antigen, there's no clumping. So let's say for argument's sake that in this sample that we've just used, let's say that this one clumps and this one clumps, but this one doesn't. If that happened, what would this person's blood type be?
A positive.
A positive, exactly. Because that what that's saying is that if we were to look at their red blood cell under a really large microscope, what we would see is that this one has triangles on the surface, the A antigens, and it has RH antibodies on the surface. That's why the antibodies were able to grab onto them, but there were no circles. So this person's blood would indeed be A positive. All right? If you're in an emergency room and need blood, the nurses don't know your blood type, is testing a fast process. Yeah, it's absolutely a very fast process. You basically get the 3 samples of the blood, put a couple drops of these antibodies in it, and you stir for about 15, 20 seconds. And just that quickly, you'll see the results, all right? Let's take a look at some pretty pictures.
Couldn't they just give you O negative though?
True, but you got to remember, O negative is not very common. And so O negative blood is very, very, it's not very rare, but it's fairly rare. And so it's very valuable. So in some cases, in an extreme case, they can give you that, but it doesn't take very long to type somebody's blood. So it doesn't take very long to be able to do that. All right. Now, notice here we're not looking at RH factor, but as we look at this, if someone, we had 2 samples of blood, put anti-A into one of them and anti-B into the other one, what would we expect to see for someone who had AB type blood?
It
Would
would
they both
agglutinate.
coagulate, just the A, just the B, or neither?
Both. They're both.
Both would agglutinate. What about B? Would anti-A agglutinate? Would anti-B agglutinate? Would neither agglutinate? Would both agglutinate?
B.
Anti-B.
Anti-B.
Just anti-B. What about A?
Anti-I.
And what about
A.
A? For O?
Oh, neither.
Neither. There you go. It really is just that fast. And the results are just that dramatic. The only thing we have to add is a 3rd well to do RH, and we can tell exactly what somebody's blood type is. Which is kind of what you guys did on your lab simulator, right?
The physio.
All right. Questions on this? All right, excellent. That is everything. We talked about this, and again, so we won't throw the RH factor in, but you definitely need to consider the RH. But let's go through it again. If you're type A blood, what did we say you could receive?
A and O, negative.
A and O, excellent. If you have type B, what can you receive?
B and O,
And if you're AB, what can you receive?
A, B, and O.
ABO. ABO.
And A, B,
O.
O, and AB. Don't forget
And AB, yeah.
receive all four. Whereas if you're O, what can you receive?
O.
O.
And that's the other reason you want to be careful with O negative. If you have someone who has O negative blood, what can they receive?
O negative.
Yeah, O negative, that's it. They are the universal donor. They can give to everybody else, but they can only receive O negative. In fact, many people who are O negative actually donate fairly regularly, and some of that they give blood fairly regularly, and some of it they will actually bank for themselves to make sure that they have blood necessary in case of an emergency. It can be a little expensive to bank your own blood, but if you're O negative, sometimes it can be worthwhile. All right, and then we talked about RH. All right, excellent. That is everything we need to know for the next exam. All right, so here is what we will do. That is all the information I wanted to cover for today. What we'll do is take one more quick break. We'll take a 10-minute break, come back at 2:50. That way, anybody who wants to run screaming from the building has the opportunity to do that. But in 10 minutes, I will happily answer any questions you guys have about the test-taking process or about any specific information for the test. All right, so any questions on blood typing before we take our break?
All right, we're not forming groups today, are we?
No, like I said, after the exam. We will be doing it on the 17th after the exam. We're not worrying about groups. We're not worrying about topics. We're not worrying about anything but Tuesday's exam. Your sole focus is on Tuesday's exam. Ignore, the only reason I mentioned the project is because I want to make sure you guys are here on Thursday. Sometimes people are a little hungover after an exam, figure, we don't do anything important on the first day after an exam. It's just introductory crap. But that's not the case here. The day after the exam is super important. It's always important to be here. You're in 431. You should know that. But it's always important to be here, but it's especially going to be important to be here on the 17th. So that's the only reason I mentioned it, but do not give any other thought to that project until you get past the exam. The exam is your focus. All right, any other questions? All right, 2:52, let's meet back here and I will answer any questions. So let me go back to our whiteboard, share that, and again, we will return at 2:52. There. All righty, let's go ahead and get started. Looks like there was one question during the break about that RH factor. Yes, that RH factor is one, can in extreme cases lead to miscarriage because it can actually, if it's damaging enough to the babies and the baby's blood, it could in extreme cases cause miscarriage. So that is one of the concerns for that. All right. At this point, like I said, I have told you everything that I think is important for the exam. So this is your opportunity to ask me questions about material that you're unclear about to help you to be successful on the exam on Tuesday. I want to remind you that on Tuesday, all we are doing is the exams. There is no lecture. We are not meeting on Tuesday. I know if you look in Confer Zoom, there does show a class on Tuesday, but again, that's just because when I set up that repetitive, weekly meeting for us, there's no way to take days out of that. So even though there shows it on the schedule there, we're not meeting, you are just taking the exams. I will be around, I will be online. So if there's any issues that you need to get a hold of me for, then you're welcome to do that. Obviously not while you're taking the exam. Make sure you scan your work area slowly and completely. Again, don't lose points for not doing the simple things right. So slowly scan your area. Again, don't have any electronic devices on you or near you, any books or anything along those lines. Stay in the center of the screen, stay will let, do all the obvious things so that you don't lose points. Like the exams in 430, somewhere between 7 and 9 is usually about the number of essay questions. And again, be the smart, sophisticated student. If an essay question is worth 4 points, should you write 5 paragraphs in that answer?
No.
No, of course not. If a question is worth 10 points, should you write 1 sentence as your answer?
No.
No, it'd have to be one hell of a
Sentence.
Yeah, exactly. Hell of a sentence, and probably a super run-on sentence for that to be the case. But for the most part, again, it's pretty simple and straightforward. That's what I like about this class. The more points a essay question is worth, the more you should be writing, the more information, the more detail I'm looking for on those. So make sure you are careful with that. All right. What
I had
else?
a question on the homework.
Yeah.
Hold on, I gotta find it though.
Okay.
It was something along the lines of like three causes of the sympathetic nervous system on heart rate or something. And I think I wrote, sorry. Okay. Yeah, so it was predictive effects of a drug that blocks the sympathetic nervous system on blood pressure and heart rate. And it said give 3 factors. I said not allow increased contractility of the heart, not allow vasoconstriction of blood vessels, and not allow the heart rate to increase.
There you go. All of those things absolutely would affect both the blood pressure and the cardiac output, how much blood was being pumped out as well. Absolutely.
Okay, I was kind of iffy on that one.
Yep, sounds smart. Sounds right to me.
What if parasympathetic nervous system is blocked?
Well, so with the parasympathetic, remember the base effect that you're going to see from that is an increase in heart rate. Remember, there isn't autonomic tone on the ventricles. Only the sympathetic innervates the ventricles. Only the sympathetic innervates the blood vessels. So you're not going to see a dramatic increase in blood pressure, but what you will see is an increase in heart rate. If you have to use, your phone as a hotspot for your Wi-Fi, you can do that, but yeah, you have to make sure it's not within your immediate space. So you have to set it far enough away that obviously you couldn't reach it from where you are, but obviously you have to be close enough to be able to use it as a hotspot. So just make sure you're putting it in a location where it's not within arm's reach is basically what we're looking for.
And you have posted all the key answers for those assignments that we did?
Yeah, the cardiovascular, the unit reviews, all of those I think are on there. I don't think I have a key for the AP, the interactive physiology thing, because that's a new assignment that I just got. I don't have a key for that. But all the other ones, the cardiac exercises and the unit reviews, those all have keys that are posted, yes.
Why would the AV node take over the SA node? Is it to regulate the heart rate?
Well, if there was so again, remember, all of the cardiac conductive cells are autorhythmic. They're all capable of firing action potentials. But the SA node is the one that fires the fastest. So when it fires, its action potential spreads to all the others. And so like a wave, it spreads through all the others. If there is a problem with the SA node where the SA node isn't firing its action potentials, then the AV node would be the cell that fires most rapidly. And the AV node would be the one that then set the wave that would spread through the rest of the cells.
Why would there be a problem with the SA node?
It could have been damaged from lack of blood flow to the area. So if there is a blood clot or something like that that damage it, decrease in blood flow to the area, some type of injury to the heart or something along those lines, you know, or congenital issues, things along those lines.
Okay, thanks.
Remember, the SA node doesn't, well, it does receive input, but the key to the SA node, like all the cardiac conductive cells, is they're autorhythmic. They don't require input. They fire their own action potential. They are autorhythmic. Remember, they don't have a resting membrane potential, so their membrane potential is constantly changing. Now, that being said, it does receive input from the sympathetic and parasympathetic and also hormones that allow us to modify the heart rate, to make it beat faster or slower as we need it. So we can modify it so it does receive dual input from the sympathetic and parasympathetic, and then of course hormones can affect it as well. But it doesn't need those things. You rip the SA node out, put it in a beaker, and provide it with all the oxygen and nutrients it had, and it would fire action potentials on its own.
And like caffeine and nicotine, they are considered the artificial pacemakers.
Yeah, so those are artificial pacemakers, things that can modify your heart rate, right? Anybody who's ever had a quad shot from Dutch Brothers knows that too much caffeine can definitely affect your heart rate, absolutely.
So they act like the SA node in a sense.
No, they modify the activity of the SA node.
Oh, okay, okay.
Yes, you should be able to redo any of the lapsters. Again, it takes your top score. And again, as I've mentioned, when you do the lapsters, because it is synced with Canvas, as soon as you complete the activity, your score is input into the grade book. But as I said, I then go back manually, and as long as you've completed 100% with 80% accuracy, I do change the grade to the full credit. So you do get full credit for that. I haven't done it for all of those. I'm a little behind with my grading, but I will catch up over this weekend. So yes, if you go back and redo the lapster, it should not change the grade, because again, it should be whatever the manual change that I did, or that way. But if it does change your grade, if you go back, then let me know and I can fix it. But it shouldn't, because it should always take the highest score, which is the one that I input. So I haven't had a problem with that in the past. So hopefully that won't be an issue. Should you be expected to do math on this exam? Yes. Right. Again, you have your whiteboard and other things. Again, it's not going to be elaborate math, but are there some equations that we've talked about? Could you be expected, if you think of the, we talked about net filtration pressure, could be something you would have to do. We talked about stroke volumes, cardiac output. If I gave you numbers for those things, should you know those equations, know how to calculate those things and actually calculate them if you needed to? Yes, absolutely. It's not going to be any, complicated math, but yes, there could potentially be some math on the exam.
Can you have our formulas with us, or we have to remember them?
I'm sorry?
Do you have to remember the formulas?
Yes, you need. Yeah, I'm not going to give you a list of the formulas. You're responsible for knowing the formulas. Yes. We didn't talk about the heart block too much. So the heart block, again, is typically a delay in the signal, primarily either in the AV node or in the AV bundle, the bundle of His. There are three main types of heart block. There is type 1 heart block, where it delays the signal. The signal always gets through, but there's a delay. Type 2 heart block is where it's intermediate. Sometimes the signal gets down to the ventricle and sometimes it doesn't. And then a complete heart block is where no signal gets from the atria to the ventricle. So I don't think we didn't talk about that too much. So I don't imagine that would be something that we would be emphasizing. We do when we get to play with the ECGs in the classroom, but since we didn't get to do it in this class, then I don't think it's something we're going to majorly talk about. So again, the format for the lecture exam is going to be some multiple choice questions, some fill in the blank, and some essay. The lab exam is all fill in the blank answers. You're going to have spaces where you write. It is not multiple choice. You do not have a what's the term I'm looking for? A terms list where you can select from to pick the correct answers. No, this is college. You are adults. I know there are some instructors who do things that way, but you are responsible for knowing this material and knowing how to spell this material. So there isn't a study list or anything along those lines. I prefer, again, remember spelling matters in this class. So, writing out the names of things, you need to write out, atrioventricular node, not AV node. So typically in the classroom, you have to write out artery and vein. You have to write out right and left. But honestly, in the classroom, I do that because it's too easy to change an A into a V or an R into an L along those lines. And so if you have to write it out from a test taking standpoint, that's easier. It's not as big of a deal on this one, but again, since spelling matters, I would prefer you write out right and left than R and L, but I probably wouldn't mark it wrong unless I'm really cranky, my kids are bothering me, I haven't had enough alcohol. If one of those things happen, I could get cranky and I could take partial credit for that. So it's always better to write those things out. And that's the other thing too. Don't lose the easy points. Right? There is nothing that frustrates me more than having somebody write popliteal artery or popliteal and forgetting to put left or right, or forgetting to put artery and vein, right? When you give me the hard part of the answer and you forget the easy part of the answer, you're still missing part of the answer and I still can't give you full credit for it. So don't miss those easy parts. Make sure you're putting left or right. Make sure you're writing artery and vein so that you're not losing the easy points. Don't get the hard part right and then forget about the easy part.
Professor?
Yes.
What was it? Shoot, what was I gonna ask? Oh, when we're doing the essay questions, if we spell it out, like say for instance the atrioventricular node, if we spell it out one time, are we allowed to abbreviate it if we follow it up in the next sentence?
Absolutely, yes, absolutely. So, and you can do that one of two ways. In your description, you can write it out once and then abbreviate it after that. Or the other thing you can do at the top of the page or at the bottom of the page, if you want to write out a key, SA means this, AV means this, right? And do that at the bottom as a key and then use your abbreviations throughout. Yes. For the essay questions, all I need is you to write it out once. And then after that, you can abbreviate it. Obviously, in a lab exam, right, you'd have to write it out, and again, but it shouldn't be the same answer multiple times, right, on an exam. So it shouldn't come up as an issue that way. Although, again, you got to remember, one of the differences is I'm not really truly writing this exam. I've written all the questions, but every single person in this class is taking a different exam. So is it possible that one answer may show up more than once? It's possible. But I usually try to avoid that when I'm writing the exams myself, but because of the random selection of the questions, it is possible that it could be more than one. So if for some reason sinoatrial node is two questions, is the answer to two questions on the lab exam, you will need to write it out for both of those. All right, hold on, I'm falling behind on the questions. So give me a second to go back. No extra credit questions on this exam. No, again, the way Proctorio works, it's not something that is easy to use. Unfortunately, there are some limitations to Proctorio. It's the same thing with the curve. The way I normally curve the grades in the classroom, I am not able to do in Proctorio. So there are what they call fudge points where I am able to do some curving to the grades. So I try to make it compatible to what I use in the class, but it's not identical to what I do in the class. And so part of that is there are no extra credit questions. There are extra credit opportunities. There was the 5 for 5. There may be some other opportunities like that. And then of course there's the Open Lab and the Science Success Center, which you can do for extra credit as well. What factors affect blood volume? So blood volume, obviously, our erythropoietin is one of the major ones. We talked about antidiuretic hormone. We talked about all of those other hormones that are involved in blood volume. Because remember, when we affect blood volume, we affect blood pressure. So basically, controlling blood volume and controlling blood pressure go hand in hand. So those are factors that are going to work together. Anything that affects blood pressure typically affects blood volume as well. When describing the function of a baroreceptor, I would be fine with saying it's a Strept receptor. They are indeed in the aortic arch, but remember they are also in the carotid bodies, where the common carotid branches to the internal and external carotid. I guess on an essay question, if you just called them Strept receptors, that would probably be example one of those okay answers that we talked about. Did we talk about that in this class? Sometimes, and again, it's again harder in this format when I'm grading this online, but even with the online, you will sometimes in the comments of it, you will see that I wrote okay. Okay is my way of saying that You are getting points for this question, for your answer to this question, but it isn't truly 100% correct. Again, I do this because what I found is that most people don't, when they review their exams, don't pay any attention to the things they got right. They just look at the things they got wrong. But since this is a cumulative class and I do need the exams to be a learning experience, this is my way of saying to you, you got the full credit for this, but it's not fully a correct answer. So that's probably what would happen with something like that. And then the command center for that reflex does take place in the medulla oblongata. That's where the nuclei that control heart rate and blood pressure and things like that are located. Yes, for both the lab and lecture exams, you will be presented with all the questions at once. So if it's 80 80 lab questions, all 80 lab questions will all appear at once, and you can go through them in any order that you want, go back and forth to them as much as you want. But again, you are still on a time limit, so I would try to go through it in a meaningful fashion, and then if you have to scroll back and forth, you can do that. But it is available all at once, and so you will just submit the exam at the end after you've answered all of the questions. So it looks like I answered that twice. So obviously you can go back because they're all appearing at once. For the exams, after they've been graded, after they have been curved, what I do is I make, because I want the test to be a learning experience, I will make the tests available for a 24-hour period of time where you can go back and review the exams, and then I will shut them down after that. So again, I want you to be able to go back and learn from them, but again, you will be limited in your ability to be able to do that. So there will be a brief period of time where you're going to be able to review it, but that is going to be it. And if you don't review it during that time, then that's on you. All right. Excellent. I think I caught up with all the ones that are in the chat. All right, what's next?
I have a question.
Yes.
Is the vascular shunt is the same as the thoroughfare channel?
Yes, and again, as we've seen before in this class, there are often multiple appropriate names for things, and both of those are appropriate names for that straight pathway from the artery to the vein that all the true capillaries branch off of. Right, and remember, in fact, there was actually a third name. Remember, we also referred to it as an atriovenous anastomosis, right? Basically, a collateral path from the arteries to the veins.
And one more question.
Yep.
Precapillary sphincter.
Yep.
They only in the arteriolar side, or also in the venous?
Oh, I see what you're saying. They would be more toward the arterial side because remember, blood flows through a capillary in one direction. So typically the precapillary sphincters are going to be located at the proximal end of the capillary bed so that they can diffuse and allow blood to flow through them all, or they can be shut, limiting how much blood flows through.
Okay.
Yeah.
In the capillaries,
Mm-hmm.
isn't it only the tunica intima that coats that one for the
Yes.
for like the elasticity of it?
Wait, I'm sorry, say that again.
The capillaries, the veins have the tunica intima only at the capillaries, the venules.
So yes, so arteries and veins have all three layers: tunica intima, tunica intima, tunica media, and the tunica externa. Arterioles, remember, only have 1.5. They have the tunica intima or interna, either of those acceptable, and kind of a scant half a tunica media. Capillaries just have one layer. They're just the tunica intima, just that simple squamous epithelial tissue and its basement membrane. Then when we get to the veins, the venules, small and medium veins all have all three tunics again. So it is the arterioles and the capillaries that are different from that. Everything else has 3 tunics. Arterioles have 1.5 tunics, and the capillary just has 1. No, it is not all right to have scratch paper on the exam. That is what the whiteboard is. Remember, part of your proctorio practice syllabus quiz was that you had to draw that blue star on that whiteboard. That whiteboard is your scratch paper, so you can use that. And again, the other nice thing about these pages with the essay questions is you can write as much as you want. So if you want to brain dump right there in the question, and then in just letters you just want to write, ignore everything above this line, and then write your answer below that, that'd be fine too. So you can use that, but you can't have a piece of scratch paper here on the table. Yes, I know you could show me the scratch paper and say, see, there's nothing on it. But who's to say that someone else doesn't hand you a new piece of paper that has all the answers on it or something along those lines? It's just impossible to be able to regulate something like that. So no, there's no external resources of any type. I had a student who during the summer had a whiteboard. She had a lap desk that also had a whiteboard on it. And so she was, she was sitting on her couch taking the test on this lap table. And then at one point during the day, she took it up, showed that it was a whiteboard, wrote on it, and then, to summarize her thoughts and everything, and then went on with the exam. And unfortunately, she got a 0 because of that. So again, you cannot have any type of external resource. That's why it's really important to have your desk and the other areas clear when you're taking this exam, because it's all about maintaining the integrity of the exam. So no scratch paper. You can use the test itself. You can use that whiteboard to put it. So again, if you have like mnemonics that help you for the lab exam and you want to have your whiteboard up so it's there as you scroll through the pages, that's fine. If you want at the beginning of an essay question, mind dump all of your information. And like I said, you don't even have to erase it. Just write a line, put a couple spaces, write a line, says, ignore everything above this, here's my answer, and then write your answer below that. And that's fine as well. So there are plenty of ways that you can collect your thoughts during the exam without having to use paper.
How it will work when you want us to compare the skeletal muscle cells and cardio muscle cells, how they work? How it's gonna work? Like everything that here is like this, but not here.
Right. So anytime, and so that's a great question. Well, let me answer Levon's question for you really first. The simple answer, Levon, is very rarely does anybody get 100% on the test. But on the off chance that, so typically in a typical class, I'd curve to what the highest grade is. If that highest grade is either 100% or significantly higher than the second grade, then those outliers I don't let affect the curve. So if there are some of those outliers over there that tend to peg the test, and it does happen on occasion, and I know from 430 we've got some really sharp students who are very successful on this test. And here, I'm actually really interested by looking at this, and I won't mention any names, but I'm very curious to see who's going to get the top score on this first exam. And many times, actually, it was really fun. Last semester, before everything went crazy in the world, I had 2 students from 430 that were competed all semester long during my 430 class, and then we got to 431, and they actually both got blown out of the water from one of Eckerman's students. So it was so it's always interesting to see how the first tests go. But yes, if you can get a 0. Yeah, if you can talk everybody into all getting a 0, then yes, it would all curve up where you all would get decent grades. But again, it doesn't curve the way it did before. So you might get a few points, but you wouldn't get an A out of that. It doesn't work that way anymore. But I won't let an outlier stop other students from being successful. Again, I don't want one student's grade to affect other people's grade. Everybody in this class has the opportunity to get 90% of the points, and every single person on this class has the opportunity to get an A. Nobody's grade in this class affects anybody else's grade in this class to be able to be successful in this class. So if everybody here gets 90%, everybody's going to get an A. I want this to be a collaborative environment where you guys are working together to be successful. So Levon, that nobody's grade is going to affect and hurt your ability to be successful in this class. All right? I felt like there was another question there that I got sidetracked from. Does somebody else ask a question?
About comparing the skeletal and cardiac muscles.
Oh, right. Okay. Sorry. Yes. I thought that other one would be faster, and then I ended up on my soapbox. So the same way we did in class, so do you need to remember what like the muscle action potential of skeletal muscle to be able to compare it to the cardiac muscle? Absolutely. But that's exactly what we did. Now, obviously, I had the advantage of drawing it. You don't have the advantage of drawing it. However, The same way you drew the star in the white box, if as part of your discussion, you want to bring up that white box and you want to draw these action potentials, you can do that as long as you are also describing them. The one thing that I want to warn you about with your drawings, and I warn people about this when we're in the classroom as well, I am not an art critic. It is not my job to interpret your drawings. Okay, so there's nothing wrong with doing a drawing to help you describe your answer to a question, but you need to be descriptive. You still need to use the words to describe it, because otherwise I'm left to try to figure out, does this person really understand what they've drawn here, or did they just memorize a picture and they just recreated that picture without understanding what it means? So every single question that I've asked on this exam is something you can answer without having to do a drawing. If you would like to do a drawing to help you to be successful, then that would be fine for you to be able to do that, but it's not required. None of the questions requires you to do a drawing. Okay? Will the 5 types of questions on the cardiovascular anatomy list show up in the lab or lecture exam, or both? Both. Again, remember, this class is one class. It's not a lab class and a lecture class. All of the material is combined together. So it is anything we learn in this class in our quote unquote lecture time and quote unquote lab time. And that time is really blurred in this when we're all online all the time. All of that material is all relevant all the time. Now, again, obviously, lab exams lend themselves more to identification. Lecture exams lend themselves more to descriptions of processes. But could I ask you to identify things or describe things on a lecture exam? Can I ask you the functions of things on a lab exam? Absolutely. I guarantee those 5 questions are questions that are going to be on the lab exam, but could I have a question, for instance, like the cardiovascular exercise? Will you have to give me the driving directions from the heart to one part of the body and back again? Is that a possible essay question where you would be using that relationship of the blood vessels to describe and answer a question on the lecture exam? Absolutely.
I'm just wondering if you can go over the blood flow for the brain, the circle of Willis.
Yeah, we can do that.
Okay. One more question before that.
Sure.
Vascular anastomosis. Am I saying that right? Anastomosis?
Yes.
Is that just for the brain and heart, or is that just more common in the brain and heart?
So an anastomosis, remember, is an elaborate network of blood vessels that allows alternate routes for blood to flow to maintain consistency of blood flow. Okay? Areas where that is especially important are areas like the heart and like the brain, where there is those tissues are highly dependent on oxygen. If deprived of oxygen for even a brief period of time, they could be damaged. But remember, we also talked about how it's really important, for instance, in joints. So no matter what orientation my elbow is in, blood is able to get to my fingertips. Right, so that is anastomosis. The arterial anastomoses, like the ones in the brain, the ones in the heart, the ones in the joints, are important for our arteries, and those are more common regions for the arteries. Veins, they're more common all over the place, because since veins are dealing with low pressure blood, it is important for that blood to have multiple routes to be able to, as easily as possible, find its way back. And as we talked about, even in the capillaries, we have some arterial venous anastomoses, connecting those together as well. All right. All right, so let's answer your first question. The important thing to remember is there are basically 4 blood vessels that bring blood to the brain. The 2 biggest are the internal carotids. So, and again, for simplicity, I will just write out internal carotid artery. Wow, that is huge. Let's make that a little smaller. The internal carotid arteries, which bring the blood up. From the internal carotid artery, there are basically 2 main paths that the blood can go. There are the anterior cerebral arteries, which carry the blood towards the front part of the brain. And there are the middle cerebral arteries that carry the blood to the lateral part of the brain. The other 2 blood vessels that primarily bring blood to the brain are the two vertebral arteries that travel in those transverse foramen of the cervical vertebrae. They fuse together to form one blood vessel. And what is that one blood vessel that they fuse together to form?
Basilar.
Basilar?
Basilar, exactly, excellent. And then that basilar splits to give blood to the posterior part of the brain, forming the posterior cerebral arteries. So this way, the back, middle, and front of the brain gets blood. Okay? Those are the main paths, those are the main routes. Does that make some semblance of sense? Right. But as we mentioned, it is vitally important that our brain tissue maintains well vascularization. Not only does it need to get enough oxygen, but if you think about what we talked about, for the appropriate exchange of materials, we need to have enough blood flowing to the area, that we have enough blood pressure, that we get that exchange that takes place in the capillaries. So we need to make sure we maintain blood flow to the nervous tissue, and that we maintain that blood flow both between the front and the back of the brain and the left and the right of the brain. To do that, we have three communicating blood vessels. The first is the anterior communicating artery. That anterior communicating artery helps to connect the right side of the brain from the left side of the brain, especially in our prefrontal cortex, so that we can maintain consistent cognitive thought. But we also need to be able to maintain blood flow from the front to the back of the brain. And so we have the two posterior communicating arteries. One of the key things to remember about these communicating arteries is while they carry blood away from the heart, right? After all, this one's carrying blood this way, and this one's carrying blood this way, and this one's carrying blood this way, and this one's carrying blood this way. These are all arteries carrying blood away from the heart. The key to these communicating arteries is that blood can flow through these communicating arteries in both directions. So if there is a decrease in blood flow to the front of the brain, blood from the back, whoops, no, hold on. Blood from the back can be brought to the front or to the side. Conversely, if the posterior part isn't getting as much blood, blood from the front can go to the back to meet the needs. So notice the key to these communicating vessels is they can allow blood to flow in either direction. So basically what we have here is a circle of blood vessels that allow us to keep a continuous blood flow and a continuous blood pressure, both to the front and the back and the left and the right of the brain. All right. Yeah, it helps to equalize the pressure, helps to maintain that blood pressure, maintain that blood flow. And this is the circle of Willis, because good old Bob Willis was the anatomist who described it. All right.
So it's circulating at all times. Well,
yes, it is allowing for the exchange of blood, keeping a consistent blood pressure and a consistent blood flow to the nervous tissue. Yep. All right. Next question. Yeah, it's kind of like a roundabout. Absolutely. Although we're Americans, we hate roundabouts. Roundabouts are bad. Even though studies have shown that they're more efficient for traffic flow. I agree, we shouldn't. But we're Americans, we do lots of stupid things. I mean, hell, we can't get everybody to agree that masks are good. You think people are going to suddenly allow roundabouts all over the place? That impinges on my freedom of choice. Exactly. All right. No, don't even get me started on the metric system. I'm still mad about that. Way back in 8th grade, we had to learn about the metric system because on Tuesday we were going metric. I don't know, for those of you at 430, you guys have heard this story. So again, 8th grade was literally 5,000 years ago for me. But at that time, and again, this tells you about the education we got in 5th grade. I mean, in 6th grade. We learned in 6th grade. Oops. There you go. Back in 6th grade, we had this sad social commentary that our teachers taught us in 6th grade, right? Kids have drugs, moms don't care much. And this was the mnemonic that in 6th grade, our teachers taught us to help us to learn the metric system. Because if you think of it in terms of meters, you have kilometers, hecameters, decameters, meters, decimeters, centimeters, and millimeters, right? Because back in 6th grade on Tuesday, we were all going to go metric, and Tuesday came around, and we didn't go metric, and nobody spoke about it ever again. But here it is 5,000 years later, and I still remember that stupid mnemonic, which shows you the power of mnemonics. All right. Off my soapbox of how stupid we are. Any other questions? All right, ECG. How many deflections do we have in the ECG? 3, excellent. What is the first deflection called?
P wave.
What does it indicate?
Atrial depolarization.
Excellent. What is our second deflection called?
QRS complex.
What does it indicate?
It says afterwards.
Ventricular depolarization.
Polarization and atrial repolarization.
Excellent. So 2 things are going on. That's why we have this complicated wave. We have the repolarization of the atrium and the depolarization of the ventricle. And then we have our third deflection, which is
Repolarization.
P wave, which indicates what?
Repolarization of the ventricles.
All right, repolarization of the ventricles. Excellent. And even though I didn't draw it that way, typically the T wave is bigger than the P wave because if you think about it, there are more cells in the ventricle than there are in the atria. So there's more change to be noted. Because remember, that's what these deflections are. These deflections are an indication of the change that is taking place. All right. Questions on that? All right, someone asked about the complete cardiac cycle. Excellent. Let's at least write out the stages. What are the stages of the complete cardiac cycle? Where does it begin? Again, it's a cycle, so really you could start at any point, but we want to pick a good natural starting point. And what is that good natural starting point?
Complete diastole.
True, although we want to be careful. Remember, the SA node is telling us what is going on in the cardiac conductive system. Right? If we talk about a complete cardiac cycle, we are talking about the mechanical events. And so what we want to start is with those mechanical events where nothing is happening in the heart. So actually before the atria contract. So at the point when the entire heart is at rest. And what do we call that stage? The entire heart was at rest.
Clinical death.
Well, so that means that your heart is never, okay, right, both the atria and the ventricles are in a state of diastole. And when both the atria and the ventricles in a state of diastole, the heart is at rest. Oops, if I spell rest right. What was occurring to the heart? There we go, that's what I'm looking for. The passive filling of the ventricles. Right? And again, technically this is a cycle, so you could start at any point, but let's start at this one just, again, if you didn't start at this one on the exam and long you went through everything, I would be fine with this, but this is a good natural starting point. Remember, when describing these, we typically want to talk about what the state of the valves and things are in this case. What is the state of the atrioventricular valves at this point?
They're open.
Open. Semilunar valves?
Closed.
Closed. Excellent. What happens next?
The atria contracts.
SA node.
So again, be careful. We don't want to necessarily talk about what's going on in the cardiac conduction system. Or let me say it this way. We can answer this question without having to talk about the cardiac conduction system. Don't mix up your essay questions. Here we are talking about the mechanical events. So as someone mentioned, we now get the contraction of the atria. Now, yes, it is the firing of the SA node and then that action potential spreading down the atria that causes that, but we get a contraction of the atria. And what does that contraction of the atria cause?
Active filling.
Right, the active filling of the ventricles. Valves change state from passive filling to active filling?
No.
No.
AV.
No, maybe. No, the answer is no. They do not. The AV valves are still open. The semilunar valves are still closed. What happens next?
Fills the ventricle to the max it can go.
Okay. How?
Pressure.
Sure, excellent. So where does that pressure come from?
The ventricle being filled, right?
No, it doesn't come from the ventricle filling. You're right. The next stage absolutely is the isovolumetric contraction. You are correct. At this point, we reach the maximal volume of blood in the ventricle. But the reason is because we can't get any more blood into it. As long as that AV valve is open, we can get blood into it. The reason we reach the maximal volume is because the AV valve closes. And what causes the AV valve to close?
Ventricular contraction.
Ventricular contraction, absolutely. So that AV valve close, again, and it's just the beginning of the contraction. It's only producing a little bit of pressure, enough to close the AV valve, but is it enough to open the semilunar valves? No. And that's why it's isovolumetric. We get an increase in pressure, but do we change the volume?
No, so it's end diastolic volume, right?
Yes. At this point, we would reach the end diastolic volume because what's happening is the ventricle is contracting a little bit, closing the AV valves. Pressure is increasing, but all 4 valves are closed, so the blood can't leave. The blood volume doesn't change. So we have that isovolumetric contraction where the volume stays the same even though the pressure is increasing. However, once the pressure increases enough, what happens?
The semilunar valve opens.
Right.
Valves.
We're able to open the semilunar valves and we release blood from the ventricle. And what do we call that stage?
Ventricular ejection.
Ventricular ejection. And again, if you said ejection from the ventricles, that's fine as well. Excellent. Does ventricular ejection last forever?
No.
No. What happens?
The pressure drops below the aortic pressure.
Right. Ventral starts to relax. And as it starts to relax, pressure drops. And you were right. When that pressure drops, the SL valves close.
No, this is isovolumetric relaxation.
Exactly. That's exactly what this is called. Again, the pressure is now going down in the ventricle enough that the semilunar valves close, but not enough to get the AV valves to open. So again, we're in the state where all 4 valves are closed, volume does not change, and if volume does not change, it's isovolumetric. But it is relaxing, the pressure is dropping, so it's the isovolumetric relaxation. Eventually, we reach a state of full diastole, and when we reach that state of full diastole, finally, the AV valve reopens. And what happens when that AV valve reopens?
Restart.
Passive filling stuff.
Yeah, we're back to the passive filling of the ventricles. And the cycle continues.
I would like
Sorry.
You would
You go
like what?
to make sure about the end diastolic volume and isovolumetric contraction. They occur at the same time, right?
So again, think of it this way. The job during this first 2 steps right here is to get as much blood as we can into the ventricle, okay? But once the AV valve closes, can we get any more blood into the ventricle?
No.
Filling.
No. So at that point, we have reached the end diastolic volume. We've gotten as much blood into it as we can, okay?
Mm-hmm.
Here, we are, oops, no, hold on. Here, we're ejecting the blood. We are getting as much blood as we can out of the ventricle. However, once the semilunar valve closes, can we get any more blood out of the ventricle?
No.
No.
No, and so there we reach the end systolic volume. And I don't like that it changed colors on me. So there. So we reach our end volumes when the valves close. When the AV valve closes and reach that state of isovolumetric contraction, we've reached our end volume, right? The end diastolic volume. When the semilunar valve closes, we've stopped ejecting blood. We're in our isovolumetric relaxation. The volume is no longer changing, and we've reached the end systolic volume.
Got it.
No. And then we subtract the end systolic volume from the end diastolic to get the stroke volume.
Yeah,
correct?
exactly. Stroke volume equals the larger number, oops, equals the end diastolic volume minus the end systolic volume, and that tells you how much blood you've pumped out. So again, if your end diastolic volume was 100 milliliters and your end systolic volume was 20 milliliters, then what's your stroke volume?
80.
80 milliliters, there you go. And again, that's the kind of math I expect you to be able to do on this exam. You shouldn't have to take your socks off and use your toes. It should hopefully be pretty simple and straightforward.
Could you call the passive filling of the ventricles just, instead of that, could you say that the atria are filling?
No, because if you think about it, the ventricles are filling during all of this. During this entire process, blood is coming back from the body into the atria.
Yeah.
During this part right here, it is continuing to go down into the ventricles, whereas here, just the atria are filling until finally it opens up and then they can fill the ventricle again. So blood is always coming back to the atria. What's different about this part up here is that because the AV valves are open, we're filling the ventricle.
Okay.
So one more important point I want to make about this question. I noticed as soon as we started talking about this, there were 2 other things that people tried to discuss. Some people started talking about the cardiac conductive system, where we generate that action potential at the SA node and it spreads across the atria, and then it gets to the AV node where it's slowed down into the bundle of His, to the bundle branches, and so on and so forth. And some other people were talking about blood flow, starting in the superior vena cava, going into the right atrium, through the tricuspid valve into the right ventricle, out the pulmonary semilunar valve to the pulmonary trunk. Those are both excellent essay questions, but are they the cardiac cycle? No. One is the cardiac conduction system, and one is the cardiac conduction system, and the other is blood flow through the heart. As I've said many times in this class, people often lose points not because they don't know the information, but because they don't read the questions carefully. I give this warning on every single test. And again, this will be my first time doing it online, but in the classroom, right, one of the things that often happens is one of the possible essay questions is to describe the action potential of a contractile cardiac muscle cell. And I have plenty of students do a great job of telling me about the slow depolarization as potassium channels close, and then this rapid calcium spike. And then all that calcium opens potassium channels, leading to this dramatic depolarization. And then another slow repolarization, to a slow depolarization, to a fast depolarization, to a repolarization, and a continuous process. And they do a beautiful job of describing all of that. And what's the problem?
They didn't answer the question.
They didn't answer the question, because what did I ask for?
To describe the action potential, or not the.
I just said as for the action potential of the contractile cell. What action potential is this?
Autoresistance.
The
conductive cell, absolutely. This is the conductive cell. Remember, the contractile cell is this one, plateau and down. Notice the student answered the harder question, and every single thing they said was correct. The problem is, did they answer the question?
So?
No. And if you don't answer the question, then I can't give you the points. I give this warning every single time. Think of it this way. If I ask you for the cardiac cycle, and you tell me how blood flows from the superior vena cava into the right atrium, through the tricuspid valve, into the right ventricle, out the pulmonary trunk, pulmonary semilunar valve, to the pulmonary trunk, to the pulmonary arteries, to the lungs to get oxygen, back the pulmonary veins to the left atrium, through the bicuspid valve into the left ventricle, out the aortic semilunar valve into the aortic arch. Does that describe one cardiac cycle?
No, it's the blood flow cycle.
Right. In fact, you would, if you think about it, you would need at least 2 cycles to do that. One cycle to pump the blood out of the right side to the lungs, and a second cardiac cycle to pump it out of the ventricle into the aorta. Again, you're providing great information. All of it is correct, and none of it answers the question. So read the questions carefully so you can answer them correctly. All right.
So if I describe the contractile cells, contractile cells AP, how it goes, and I mentioned that how different it from the skeletal muscle, but I wasn't asked about it. So it means
okay. So here's the thing that I would say about that. Obviously, the goal is to answer the question. If I just ask you to describe the action potential of the contractile cell, and you talk also about the skeletal muscle action potential, and everything you say is correct, I'm not going to ding you points. However, if you say something wrong about the skeletal muscle action potential, even though I didn't ask for it, then I'm in that case again that we talked about, where I'm left to interpret your answer? Is it that you truly understand this information, but you made one little mistake while you were typing? Or were you just mind dumping everything you could possibly think of onto the page in hopes of being able to get points? And like I said, I don't interpret answers. So if you spend a half an hour talking about some topic that is related, but not directly part of the answer, and it's all correct, I'm gonna frown a little bit, but I'm not gonna ding you points. But if any of it's incorrect, then I can't interpret that answer properly, and so you won't get full credit for that.
Okay.
Yeah, so if I asked about the cardiac cycle, I want those stages that we just talked about, right? Passive filling, active filling, all of that kind of stuff. Yes, there absolutely will be compare and contrast questions, absolutely. And again, when you're doing that, make sure you're comparing apples to apples and oranges to oranges, right? If you're comparing an apple and an orange, and you say that an apple is red and an orange is sweet, that is not a direct comparison. And remember, a comparison is not to tell me everything you know about an apple and everything you know about a orange. It is about direct relationships between the two of them, right? Compare, this one has a long refractory period, this one has a short refractory period. That is a comparison, right? Saying this one is in skeletal muscle and this one isn't a comparison. I will try to get the lectures up like I always do as soon as I possibly can. I think there was another question that I missed. Oh yeah, so the pressures. So any other questions on this before we move on to the next one about the pressures? All right, let's do this simple. How do we calculate the net hydrostatic pressure?
The pressure in the arterial minus the pressure in the venual.
No, it's
No,
the
close.
pressure in the what is it? The hydrostatic pressure of the blood and the hydrostatic pressure of the, or subtracting the hydrostatic pressure of the interstitial fluid.
There you go. So the blood, or you can think of the capillary, the hydrostatic pressure of the capillary, and from that you subtract the hydrostatic pressure of the interstitial fluid. Excellent. How do you calculate the net osmotic pressure?
Osmotic pressure of the capillary, and then subtract the osmotic pressure of the interstitial fluid.
Excellent. Oops, I spelled fluid right. And then how do you calculate the net filtration pressure?
The hydrostatic pressure minus the osmotic pressure.
Right, the net hydrostatic pressure
Net.
minus the net osmotic pressure. And remember, when we're talking about this net filtration pressure, a positive number means
It's an outward force.
Outward force. And a, therefore, negative pressure means inward force. And remember, if it's an outward force, we say it is involved in filtration, where if it is an inward force, it is involved in reabsorption. Okay.
Can you screenshot the?
Nope. When I write things out like this, so pictures are things that are screenshotted and saving, but when I'm writing things out, those are things that you should be writing as well. Tactile learning is important.
So the
This is not new. This is all in the lectures too. So you actually already have it all printed out for you.
So the pressure in the arterial minus the pressure in the venual is called what?
Okay, so it's, well, so the short answer to your question is what you've given me, what you're describing is the pressure gradient. And that pressure gradient, remember, is what allows the blood to flow. Because blood flows from a high pressure to a low pressure. The key, and I think where you're getting confused, is that if we do this calculation, if we calculate the net filtration pressure, what we saw is that it is different at the arterial end and the venual end. of our capillary.
Yeah.
So when we did the calculation, we actually did the calculation twice, once at the arterial end and once at the venous end, to see that actually in an actual capillary, at different ends of the capillary, different things are happening. At the arterial end, we're getting filtration, and at the venous end, we're getting that reabsorption. So that's why we did it twice, to show that in a capillary, it really is an exchange of materials. It moves in both directions.
Okay.
All righty. Anything else? I think we've covered dang near everything, which is awesome. But any other questions? Everybody's brain finally full? All right. Well, what I will remind you is that if you come up with more questions, I'm always good at responding to emails, but especially on exam weekends. On exam weekends, I try to be a little bit more attentive to the emails and try to respond a little bit more quickly. Obviously, you've got real life things and kid things to deal with and things, all those things along that. So I'm not sitting on my computer all the time, just most of the time, because I hate my family and I'm trying to avoid them. But I will try to do the best that I can to respond to you as quickly as possible when you guys have those questions to help you prepare for the exam. All right. Any questions on any of that?
The slow depolarization of the conduction cells is called the pacemaker potential. Is that the AV delay?
No, the delay in the AV node, remember, is because the cells are very small and very thin. And as we talked about with blood flow, and electricity moves a lot like water, a lot like fluid. So when you have small cells, when you have very narrow cells, you have an increased resistance. And that increased resistance slows the signal of the action potential down, so that it takes more time for it to reach the ventricles. That gives more time for the atria to contract before the ventricles contract. All right? All right. You guys have a happy and safe, careful weekend. Study hard. And I will see you guys on Thursday. Again, I will be on the computer on Tuesday. If everything goes well, I won't hear from you. You won't hear from me. So hopefully everything goes well. And I will not see you on Tuesday. So I'll meet you back here on Thursday. And have a good week. Have a good weekend. Study hard, everybody. And like I said, if you have any questions, please let me know this weekend. All right. Take care. And we are done.